Hi, everybody. A lot of people are joining right now. We'll give it one more minute and then we'll get started. Fantastic. So we already have 50 attendees. We expect a few more. So we're really pleased. We really have learned a lot from the online forum. It gives an opportunity, especially for rare disorders, for people to join from wherever they are. And so we're getting, I think, a lot better opportunities to get the message out. My name is Joseph Buxbaum. I'm the director here at the Seaver Autism Center. And welcome to this FOXP1 Day of Learning, something that we plan to do on a regular basis. And I can just advance my slides. And I do want to say hello and thank you from all of the people at the Seaver Autism Center. We do a lot of work in genetics of autism and rare disorders, and you'll be hearing from some of the speakers, but there's actually a very large number of people working on some of the projects you'll hear about. So we are very happy to have organized this and to host you and to have some very important guests from outside the Seaver Center. Within the Seaver Center, Pilar Trellis will be speaking, who works on the clinical manifestations of the syndrome. Nan Yang will be speaking, who works on the stem cell models of the syndrome, and Anna Kostic will be speaking, who works on stem cells and also translatable biomarkers. But as you can see on the right, there is a large group of people that are behind these projects, and I want to thank them for the work they're doing and for providing those results that we can present to you today. And the concept that I think is really kind of emerging in rare disorders is really, I think, quite powerful, and one of the areas where there really can be a lot of partnership between all the stakeholders, between academic laboratories, clinical laboratories, family foundations, NIH, biotech, and pharma. So the idea that we and others endorse is that if you, for example, have a molecular target, or you have something that, in this case, it would be a gene, FOXP1, then you can create, you can kind of learn about that gene, and you can create pipelines for drug discovery and development, DDD. And as part of that, as you will hear, you can make human neurons in culture that actually have the mutation or the gene mutated of interest. And you can learn not only about biology, but they also provide the opportunity to develop these cellular assays for screening, for drug screening, for example. And they can be a kind of a part of your process to say, should we go forward with this treatment or this drug, gene therapy? And then, as you'll also hear from Jenna, we can now knock out genes in mice. We've been able to do that for quite some time. But again, you have a model that you can learn something about the biology. You can possibly even get translatable biomarkers. So for example, if there was an EEG abnormality in individuals with mutation and in the mice, then maybe that's a biomarker that you can see. If you can improve the EEG abnormality in the mouse, you'll get also, you can predict positive outcomes also in clinical trials. And again, the mouse itself can be used as a go, no go, right? So if you found something interesting, you did a screen, you found a possible lead compound, it works in human neurons in culture, and it works in vivo in mice, you take it forward, and if it doesn't, you might stop, right? And so the final step in this pathway would be clinical trials. So the idea of starting from either agnostic high throughput screens, which is the way pharma used to work, or gene targeted therapeutics or protein targeted therapeutics like FOXP1. You make assays, you screen, you look at them in neurons, you look at them in rodents, and then obviously there's a lot that happens over here, which is you prove that it's safe, and you do clinical trials. So where pharma and biotech come in really are, they're the experts on high throughput screens. And they're also the experts on medicinal chemistry. There are some obviously great academic sites that do that, but on scale, it tends to be industry. So this is why it is such a perfect public-private partnership. And obviously, especially when we're talking about a rare disorder like FOXP1 syndrome, the families are the ones who really are behind the movement of this whole process, right? It is all done for the families, and and the families are very good about reminding us that this is urgent and we should be moving quickly. So that's kind of the context of how many people in the field are thinking about rare disorders. And the agenda today, I'll just show it here, but I will introduce each person but not come back to the slide. So I mentioned Pilar, we're talking about the clinical aspects, Nan about stem cells, human nerve cells in culture, Anna about organoids in culture, Saskia will talk about a clinical survey that she's put together, and Genevieve will talk about mouse models and some cell-specific findings in early development. But the first speaker or speakers are Carl Whitney and Gail Clark. Carl is the co-founder and co-president of the International FOXP1 Foundation, which something that I know he's been working on for quite some time. I think it's an important, huge step for the families. And he'll also be presenting with Gail Clark, who is the member at large of the International FOXP1 Foundation. And Carl has really, without question, is responsible in a very significant way for the Seaver Center beginning to study FOXP1 some years ago. And we've had a great partnership with him, and he's on our advisory board and associates board, excuse me. And he can take all the credit for anything that's going on in the Seaver Center. So I'm going to unshare. I forgot how to do that. And I will turn it over to Carl and to Gail.
Joe, thank you very much. I was just having a little bit of technical difficulty sharing my screen and getting myself off mute at the same time. So let me try that sharing again.
But what I said recently was in about a year from now, I'll finally have mastered Zoom.
So can you all see my slide presentation?
Yes, it's
okay.
It's in. You could probably switch to full screen mode. Right now it's in presenter mode, but
or you can swap the display if you have two screens, and then we'll see the other screen.
There it is. Take it away.
Okay. So Joe, thank you very much for the introduction. I'm sorry about the technical glitch there. I wanted to take a few minutes just to introduce the foundation, explain who we are and what we hope to accomplish. So as Joe mentioned, I've gotten to know the folks at the Seaver Center over the past few years. And one of the important things that we did together back in 2019 was to plan out and put on a really nice parent researcher conference in 2019, where a lot of the parents and families got the chance to meet each other for the first time. This was a really important thing for the community. And around that time, we started to talk about founding or getting together a foundation to help push community building and other things forward. So we finally got enough critical mass and momentum in the beginning of this year, in January 2021, to actually formally and legally establish the foundation. We have US 501c3 status, which sets us up as a nonprofit to accept donations on a tax advantaged way. And we were set up intentionally to be international in scope. So we have a board of directors that comes from a number of different countries, and we have country ambassadors that are in a larger number of countries around the world. And we all work together in establishing the foundation and trying to do what we're setting out to do. So just in the big picture, our goals are to build that community, to continue to strengthen our connections across families, to help support each other and the individuals that have FOXP1, syndrome, to strengthen communication, as I mentioned, and just to like simple things like sharing knowledge and new treatment ideas that we've heard about, and important research updates as well. And then the third sort of leg of the table here is to establish a registry. And this is really important from our perspective, just like from the simple basis of figuring out who we are and where we live. And even, what kinds of age categories do we have represented in the FOXP1 community? And that's very valuable for really basic things like helping home in on a subpopulation that maybe a researcher wants to work with to learn more about the syndrome, or collecting information about the symptoms of the syndrome on each individual basis, and collecting updated information over time to get a better sense of the natural history of the syndrome. And I think that will be very important as we go forward to help really well characterize the syndrome and understand more about where people are going. That will help us understand the prognosis and developmental trajectory for individuals. And then as we continue to grow, we want to exploit that 501c3 status to fundraise and to eventually support research. Now, I don't believe that Gail has speaker privileges, so I'm going to go ahead and cover slide 2 of our short presentation here and just run into a little bit more detail what we're doing now and what we hope to accomplish in 2022 towards those bigger picture goals. So we're already doing valuable things like running monthly Zoom meetings where we get a dozen or so people every month get together for open-ended discussions. And that's proving to be really valuable just for support and connection. And the community is growing very rapidly. So those dozen or so people that show up every month, a lot of them are newly diagnosed families, and they have lots of questions, and it's a great way to share knowledge. We turned some of that knowledge into a monthly newsletter that we've been putting out now for a few months. As I mentioned, we have ambassadors, and the goal of this is to have folks in each country where there is a significant number of identified individuals to help build local communities and to help, share knowledge in local languages, because a lot of us, especially in the US, do not speak other languages, and it makes it stronger and easier for us to establish a global presence through the ambassadors. We are currently planning another parent researcher meeting, most likely in the New York City area. We're having some trouble finding a good location because of the pent-up backlog for weddings and things like that from 2020 and 2021. So we may actually run that meeting virtually in 22. And as I mentioned, we have this registry goal. We've identified the platform and we plan to go live in early 22. We're very excited about that. And the other thing that I'm very excited about is that we've been able to establish a scientific advisory board, including representation from the Seaver and in the form of Anna Kostic, and also Jenna, who's speaking today, is a member of the SAB as well. And our first meeting is now planned for November. We look forward to working collaboratively with the SAB to help us chart out that forward-looking direction for the foundation. It's a very exciting time here. Last thing I want to say, just to shamelessly plug, is that the board of directors is, we are a finite number of people. We're about 8 folks. We can't do everything. And we are always looking for volunteer support. So reaching out to the folks that have dialed into the webinar today, if you are interested in helping any of these activities that I've talked about here, please contact us at that email, info@foxp1.org. We're particularly looking for volunteers to help us implement the registry. That's not going to be an easy, light lift. And the more folks we can have to help us, the faster we will achieve our goals. And I'll wrap it up there and just thank you all again for this platform and opportunity to speak. I'm very much looking forward to the presentations today, and thanks again to the Seaver for organizing. So I will stop sharing now. I will try to. There we go. Okay, back to you, Joe.
Oops, thank you. Whoops, I'm having some technical difficulties. There we are. Thank you, Carl. And I'm sorry, Gail, we probably should have put you on speaker mode. So I will also remind people that there's a Q&A, and there's a Q&A button at the bottom. There's also a chat. The chat should be disabled, but apparently it's not. If you put questions in the Q&A, I, as moderator, will bring them forward. And maybe since there are a couple of points, I will just say that, Carl, we have somebody who, you can see this actually, because it's you're on the list, right?
Yep.
Well, okay, this is a Jeff,
I'll respond to that, Joe. It's sort of directed at the foundation, not the Seaver. And just to clarify for everyone, of course, the International FOXP1 Foundation is a separate organization from the Seaver Center. We are clearly working collaboratively, but the communication piece in these newsletters is on the foundation. So, Jeff, I will write you a response.
Well, Jeff, I also want to say that I apologize if the Seaver Center has not responded well. There's no question that the past 18 months has been very hard for us, and I wasn't so good to begin with. It only got worse. But Jeff, if you'd like to reach out to me or anybody else and tell us how we could be helpful, that would be fine. So again, if you use the Q&A, then I can moderate, and we'll be getting this question a lot, I'm sure. Karen also asked about how to join the monthly Zoom meetings and get the newsletter. So maybe Carl, at the end, we can send that information to everybody who signed up, if you can help us with that.
Yes, I'll get you the information and you can distribute it. That's the most efficient way. Thank you.
All right, so the next speaker is Pilar Trellis, who is assistant professor here in the Department of Psychiatry and in global health as well. Pilar is a child and adolescent psychiatrist with vast expertise in neurodevelopmental disabilities and really also in rare disorders and perhaps most importantly in complex developmental disabilities and how to make accurate diagnoses in the context of complicated presentations. She's very interested in kind of bridging research to clinical care, had an important role in a very important clinic here at Mount Sinai, and is really focused, I think, on monogenic forms of these developmental disorders because there is a translational potential and the clinical phenotype is pretty much unknown until you bring in a bunch of individuals with these rare variants or the rare mutations and study them, and then you can think about, as we talked about earlier, disease-specific treatments or syndrome-specific treatments. And she's really been one of our leads in the FOXP1 clinical characterization of the FOXP1 syndrome. So Pilar, I will turn it over to you.
You can hear me well, right? Let me share my screen. Sure. Okay, so it's very nice to be here. I wish I could be seeing all of your faces rather than only mine, like we used to do in the old day, which I hope will come back soon. We decided to focus on the very complex behavioral phenotype that we see in kids with FOXP1 syndrome, because we think there might be opportunities for intervention that you could probably, and I'm hoping some of this you can kind of bring back to your physicians and maybe figure out ways in which you can implement some of the things that we are going to talk about today. So just as an outline, I'm going to speak very briefly about kind of where the field stands, where kind of what we know about neuropsychiatric and medical findings. Then we're going to delve in a little bit more and kind of dissect many of these symptoms, and we'll share with you what we've learned from our clinical studies. I really want to make a point about kind of bridging what we know, even if it's like a couple of recommendations in a way that can really inform clinical care. And finally, I'm just going to mention a few things in terms of future directions, and I hope that will serve to also inform families what we are trying to do and where we would like to go as this continues to move. So these are many of the kids that we've met, who are a bunch of incredibly charming individuals, but that, as you probably know, can present with very complex and sometimes challenging behavior. So ever since the syndrome was described, we've known, which has been like in 2010, I think was the first time, we've known that these kids present with global developmental delays. For most of them, that ends up translating into cognitive delays and what we call intellectual disability. There's a huge component of speech and language deficits with motor and coordination issues. And then something that we see a lot in FOXP1 is that they present with this kind of complex set of behavioral symptoms that unfortunately can get them in trouble and can make challenging, kind of joining like educational activities, and many times for parents, just kind of parenting, because it's the type of thing that would get someone to maybe do or say the thing that they don't want to do or say at the time. Very briefly, this is FOXP1, and it's not the point of my talk to talk about kind of the science behind this, but I do want to mention, as a clinician, for me, it's very important, and when I talk to parents, just to explain, where are the symptoms coming. So FOXP1 is a transcription regulator factor, and when I think about it, I think about transcription regulators as kind of the architects of our bodies, are the ones that say, okay, this is where this should go and how it goes and how you regulate it. So in many ways, and it's a protein that is very much expressed in the developing and adult brain. So there's a huge role in how things are kind of organized, let's say very broadly. And one thing that we notice is that a lot of the mutations and the variations that we see, they kind of cluster in this area, which is the binding domain to the DNA. So that is kind of important. This comes from a study from the first group of kids that we published on. Dr. Seaver and the Rebase really worked on this. And when we were thinking about, okay, what can we add as we are moving forward, there were a few things that were important, and I'm just going to highlight them. So ASD symptoms was something that from the beginning, people were saying these kids present with a lot of like autism symptoms. What we noticed was that despite the fact that they did present with features of autism, and I'm going to dwell more on it, it was really a smaller proportion that actually had autism kind of like fully, as we described it, like kind of typical symptoms of autism. These were kids that might be maybe struggling with friendships, or maybe had some of the features of like repetitive behaviors, but on the day-to-day, they really had a lot of social strengths, and that's important. The other area was obviously cognitive abilities and how that was impacting what we call adaptive functioning, which for me as a clinician is in so many ways much more important. So cognition is an aspect, or kind of what you can in theory do, but then how you're able to use and utilize and access your skills in everyday functioning in everyday life in so many ways is much more important than the actual kind of number of an IQ. And I want to just mention here, and we'll talk a little bit more, sometimes in kids with complex neurobehavioral profiles, there's a little bit of a divorce between these numbers. If you get someone that is quote unquote like typical, these numbers look pretty much the same with the numbers over here. But when you have someone with a complex neurobehavioral profile, it affects day-to-day functioning. So there can be a little bit of a divorce. And then the other thing then, as a child psychiatrist, of course, I'm interested in this, but also because I know this can get you in trouble, and it can create can diminish like the ability to enjoy life, which is really what we want for everyone, is that a lot of these kids were struggling with anxiety, kind of compulsive behaviors, and a lot of inattention, kind of ADHD-like symptoms. And that is a terrible combination, because when you put them together, especially when you have a kid that has a hard time communicating, can lead to what we call externalizing behaviors or aggression. Very briefly, these are some of the most common medical features. And the only thing I want to say is, as you can see, they're like kind of everywhere. A lot of it relates to things that are probably secondary to also like kind of an atypical brain development, like hypotonia, even like the ocular abnormalities. But because it's a transcription regulating factor that is everywhere in the body, we've seen medical issues in other areas. And this is probably for another day of learning, but I just wanted to put it here. So kind of moving forward and what we've seen. When we decided, okay, so we know this about FOXP1 very consistently. We've been seeing it since it was first described that these kids present with these symptoms. What can we do to really look at it more thoroughly with the instruments that we have? How can we like start dissecting and saying, okay, these kids maybe are having trouble for A, B, and C, and how can we assess it? So if anyone has come through our center, we really meet with the children and the families for 2 or 3 days, depending. And then we do a number of clinician administered instruments. And then we also spend a lot of time talking to the parents. And then we also give them a number of questionnaires. This is the group that we've seen here today. It amounts to 22. Of course, there are other cohorts, and the original paper by Seaver et al. Included kids that we didn't see here that were seen at Washington University, but some of their findings are very similar to what we've seen. Most of the kids we've met are actually pre-puberals or are in the childhood age. So I think what I'm talking to you today, really, I think we can say kind of consistently, this is what we are seeing and observing, and this might be more usable, and we need to learn more about what happens later in life. But at least for these children, definitely until early adolescence, we can have a good sense. In terms of the mutations, this is a similar graph, and just in the interest of time, I'm not going to spend a lot of time, but you can see that the distribution of the alterations in FOXP1 that we have was very similar to what had been described before. This is a graph that shows cognitive and adaptive functioning. So again, because I know there's a lot of parents, and I really want to make sure that this kind of sinks in and you can kind of tie it to your child. So cognitive function is like the IQ, this number that you get. That adaptive functioning is kind of also a number, but it's a different type of number. It tells you what you can do with the abilities that you have. And it's really more a reflection of how you do in day-to-day function, okay? This number is important, of course, and it kind of gives you and tells you what a person kind of can do, but I think that this number in so many ways is much more important. So in terms of cognitive ability, we saw, and we've been seeing what we've known for a while, kids have anything from, in some cases, this bell curve, this represents the general population, okay? So we get like 100 people from the general population, and we kind of plot them here. This is how it will look, okay? So anything that goes below this is kind of less than the average, and everything that goes above is kind of above the average. So we see that kids, even some of them are in kind of in the mild, and what we call here mild intellectual disabilities, kind of up to here. So they have cognitive deficits, but for most children tend to be kind of in the mild case. And then when we look at adaptive functioning, kind of the adaptive composite is kind of what you would expect, but there's a couple of things that we observe. So things like socialization and daily living skills seem to be like a relative strength, okay, for many of these children. And I think this is important because this is something that you can capitalize on with the right supports. Something that really most of the kids struggle was communication, and I think that's something that you know about. And very recently, the group by Ruth Braden published a large cohort that I'm going to mention a little bit about because I think it's complementary to what we see. So what do we see here? We see here, like if you see this figure below and you can kind of remember where they are, you can see that most of it is like a little bit towards like the right, but then if we go to the other group, most of the communication ends up going to the left. So this is something that kids really struggle with. These are measures of receptive, expressive, and written communication. Most of the children had some written abilities. All of the kids that we saw, except one that was very young, Sam, had an ability to communicate. And in the larger group by Braden et al., they also saw the same, that most kids have some form of verbal communication, or even when they're minimally verbal. So that is important. There's a lot of questioning about if receptive and expressive language is better. I think it's very hard. Different studies have shown different things. I think for the most part, it kind of goes hand in hand. Each of these dots in this figure number C represents one child. The blue one is the receptive language, and the light blue is the expressive language. And that you can see for the most part, they are hand in hand. So that's just kind of something to think. So, speech and language and communication is kind of central and a key feature. And this is what they conclude. This is what this group in Australia concludes. So severe speech impairment is a distinguished feature of FOXP1 disorder. And I want to read it from here because I think this is important. So what they say is FOXP1 related disorder is characterized by a complex speech and language phenotype with prominent dysarthria, broader motor planning and programming deficits, and a linguistic-based phonological errors. Diagnosis of a speech phenotype associated with FOXP1 will inform early targeted therapy. And another thing that I find here that I think it's very, very important for you parents is that although most of the kids, all the kids have dysarthria, which is this difficulty articulating words and being understood, very, very few were receiving specific interventions. And that is important because when I meet a lot of the kids, not just with FOXP1, but many children with has like monogenic forms of neurodevelopmental disabilities, the parents can understand them very well. But if the other adults and the children cannot understand them, even if they have a lot to say, A, it can be frustrating, and B, it's just going to affect their ability to kind of go through life. So that's another thing. And then these things that talks about like planning and programming talks about like executive function deficits. And I want you to keep that in mind because we're going to go back to that with some of the other things that we found. This is more of the same, and kind of again in the kind of the interest of time, I'm going to go through it. I just wanted, if you have access to this and maybe through the foundation and we can share this with you, you can look at all of these different things. And I think you will be able to maybe identify where is your child and see that a lot of these things are shared by many of these children. The next part that we wanted to look at more deeply was this idea of autism. So we knew, and we had started meeting kids who are kind of an autism center. That's like our kind of area of expertise. Okay, these kids have, features of autism. Some people are even diagnosed them with autism, but when we meet them, they, most of the children we meet, they can socialize. They have like good social emotional reciprocity. Their nonverbal communication and great. There's this like, this wonderful thing. But most of them, if you see here, we're having a hard time developing friendships. And the other thing and maintaining friendships. And the other thing that we were seeing was that they have a lot of kind of repetitive behaviors, but repetitive behaviors that we typically see in kids with autism. So we can see a child that has kind of repetitive behaviors, but the repetitive behaviors that kids that do not have autism look different than the repetitive behaviors of kids that have autism. And there's something about FOXP1 syndrome that made us realize that their repetitive behaviors look kind of autistic, and that maybe can do with like sensory symptoms, and I'm going to talk about that. And we can have a lot of thought behind it, but it's there, and that can also interfere with life. So consistently with what was reported before, about a quarter of the kids that we saw had kind of what we call like more classical autism. A number of them in this standardized instrument, the ADOS and the ADR, which are instruments that we use that help us kind of tease apart and kind of find the different ingredients of this like social interaction and repetitive behaviors. Kids were showing them, but when we were coming together as a group of clinicians and saying, okay, maybe this child didn't make great eye contact during the evaluation, but you know what? I think it was because he was very anxious or he was inattentive. And I think that's why maybe he doesn't have autism. Those things are important when you assess these children, when you have like multiple eyes, and you can actually think about things in a more broader way, because you really want to know what you're treating if you want to help someone. In terms of the diagnostic classification, this graph is kind of difficult to explain, but it's difficult to explain because I think it is a reflection of what kids of the very complex neurobehavioral profile of FOXP1. And I think that's why sometimes doctors and even professionals that are working with these children kind of get lost in the symptoms. So let's orient us. So we divided the kids that we saw, the 22 kids that we saw. Sorry, there's 21 here because one of the children was too young to include. So we have our kids with autism, and then we have our kids that do not have autism, okay? And then we have this measure that is kind of it's a good instrument, it's a good number to tell us how severe the autism is, okay? And then we put them in order, and the pink, that like stronger pink are like more severe, that like stronger blue is less severe. So we kind of order them from least severe to more severe for both no ASD and less ASD. And generally, if we would do this from, for kids that do not have these kind of very complex behavioral profiles with, a little bit of anxiety and ADHD and speech issues, we would kind of see this kind of level of shading, right? Going from like blue all the way to like pink. But what we see in the kids that do not have autism, right, is we see a lot of like pinks in there and then symptoms that kind of do not make sense. And I think, I don't want you to dwell too much on it, but I think if you can take something from this, is that this just reflects how complex it can be and how difficult it can be, like in clinical practice and in day to day, to really figure out what is the most important and what's to treat. This graph only shows that the kids that have autism are the ones that have more difficulties with socialization, but that's kind of what we know. And this is the results from the sandwiches and clinician and administered and kind of parent questionnaire in terms of sensory symptoms. And this is what was more relevant. So one of the things that I think that I look at this, and when I think about the kids that I've met, maybe about those repetitive behaviors and some of the things that might get them difficulties. I'm sorry, for whatever reason, okay, this is not collaborating. Okay, is that, so in this line, so again, we have this like bell curve, okay? And this is where if we would just get like 100 kids that have typical development and we'll put them, this is where their scores would go, okay? And then if we go towards this direction, towards like the 8, it's kind of worse. And then towards here, it's kind of better. So we see that kids with FOXP1, for the most part, compared to typical kids, they kind of have more sensory symptoms in these different domains. And then we compare them to kids, just like also like a norm population of kids with autism that have kind of this classical autism. And we see that some of the sensory symptoms are more typical of what we see in autism, except for this idea of sensory like seeking behaviors, where these kids with FOXP1, individuals with FOXP1 syndrome seem to do worse. And if you think about your children, you might relate to this. You might have seen more of this in their behaviors. Okay, then just to kind of wrap up, I hope I'm still on target with that time. So another issue, and just going back to these figures with the different colors, with the pink and the blue and all the little squares, which really kind of tells you, okay, this is, when I look at that, I'm like, oh my God, these kids, their brains are just really complex, right? They have all of these strengths, but then they have these things that make it hard for them. But there was something that was very consistent and that we're seeing since we were like meeting all of the families, right? One was that they presented with what we call symptoms of ADHD, so attention deficit hyperactivity disorder, which is, you know, it gives you executive function problems and it gives you difficulty socializing and learning in a way very differently, like let's say autism. So that was one. The other thing that parents were consistently reporting and that we were observing as clinicians was anxiety. And then many of the parents, and then we saw it here in kind of where we're trying to administer an assessment for some children, they would get aggressive. I want to leave aggression to the side for a little bit because I really think this is more like a response to some of the other things that are seen, okay? So what we did this time is, okay, so the instruments that we have that we normally would use to assess like ADHD and anxiety, in kind of a very objective way, these questionnaires that we give parents, they many times don't seem to perform so well in kids that have these very, very complex neurobehavioral profiles. So we really need to rely on our own kind of clinical expertise in what the parents are reporting. And when we kind of say, okay, this is the number of kids that we see, this comes from saying, okay, you know, 2 independent clinicians that met the child through a number of hours and talking to the family felt that the child or thought that the child had ADHD or had anxiety. And on top of that, the parents reported it as the most kind of concerning or kind of the first concern that they had. So as you can see, a big chunk of the kids have ADHD, almost half of them had anxiety. And then there's a group that had aggression, which I think really could be a consequence of maybe not really treating the underlying other issues. So because anxiety and ADHD together is kind of a time bomb for a kid to really have a really hard time regulating their emotions and maybe engaging in aggressive behavior. Again, especially if they cannot communicate their needs or people are having a hard time understanding them. Again, another of these figures. So we put here all of the kids that clinically we had said, okay, these are the kids that have ADHD, ASD, anxiety, and aggressive behaviors. The ones that have the little asterisks were the ones that were receiving pharmacological treatments for all of these conditions. Two things that I want to mention here. A, as you can see here, there's a lot of maybe the scale for attention can have some correlation with ADHD, but then everything else really didn't perform so well. The other thing is that despite the fact that there were an overwhelming number of kids with ADHD and anxiety, and medications in these conditions can help, and specific behavioral interventions can help, few of the children were actually getting them. And we'll talk a little bit more about that, and I'm sure you'll have questions about it, but I think those are very objective and concrete opportunities to maybe help and treat some of these children, and of course, all of your family. Finally, this is a graph that I wish we had more kids, and hopefully we will be able to get more. So this is an instrument which is called the brief, and it looks at executive function, and executive function is what we all wish to have like perfect. Is what will allow us to like engage in multiple different activities and, you know, be planned and organized and shift from one activity to the next, and be able to kind of get a good picture of what's going around us and then interact with certain people, or maybe inhibit, like if we don't feel great about something, kind of inhibit what we're about to say and then listen to another person. So it's this like wonderful and very complex skill that obviously you develop as you get older, but kids that have ADHD, autism, and FOXP1, you know, they have issues with executive function. And we plotted it like this. We went through many diagnoses, and this is the ones that we have. So the executive function deficits of the kids with FOXP1, at least in this very small group, it resembles a lot, or has, for the most part, the same distribution of what we see for kids that have ADHD combined type. And again, this is important because there are specific things that we could do for that might help your child. So finally, and I hope I'm not too in with the time, in terms of implication of clinical treatment, I think that, you know, what we've seen is there's a huge range of cognitive and functional abilities. As I mentioned, sometimes there's a little bit of a divorce. So a kid that might have like a higher cognitive ability and maybe their functional abilities are a little lower, or the other way around. And a lot of that has to do with all of these other symptoms of anxiety and executive function problems, and, you know, daily living skills, and impulsivity or hyperactivity, how all of those other things are interfering in someone's ability to really access those skills that they have. Speech and language deficits are central, and they should be probably started very early and aggressively. And dysarthria seems to be something that needs to be addressed from early on, because that would really help a kid be able to communicate and be understood by the people around them much better, not just kind of their close family members. And then, well, we see a lot of motor and coordination issues. All of the children that we met, they were in comprehensive programs. I mean, these vary widely depending on where you live, which even within the US, within the same state, within New York, it may be different what you access in your city than in Albany. But, you know, in theory, like comprehensive educational programs for kids that have developmental disabilities should be equipped to support these kids. And at the end, it's less about the label and more about what will help the child. I think like speech, occupational therapy, and physical therapy have a role and hopefully should be started early on. Kids with FOXP1 have a lot of cognitive skills in the sense that they can engage in cognitive behavioral therapy. So maybe early on something like ABA, which is just looking at specific behaviors and really not helping the child learn about their emotions and how that is affecting others and themselves. Might be difficult, but I think as they go older, we can actually start working with cognitive behavioral approaches, and I think they could be good candidates for that. And then the last thing, and as a psychiatrist, of course, this is kind of my area of expertise, is pharmacological treatments. So there's two conditions that I think we really need to explore, and you as parents need to explore with your physicians and providers, and that is the treatment of anxiety and the treatment of ADHD. So anxiety respond to behavioral interventions and CBT approaches. Sometimes the symptoms are very overwhelming. A number of the kids that we met here were on treatment for anxiety, and they were doing well at relatively small doses of SSRIs, which are medications that are widely available, and another type of medicine, which is called alpha-2 agonist, which is kind of an off-label prescription, but anyone that works with children with disabilities, or just with children in general, are very familiar with these medications. And then in terms of the treatment with ADHD, we do know from many, many years of studies that pharmacological interventions are central and key in the treatment. So when I diagnose a child with ADHD, not just someone with FOXP1, just in general, I always explain to the parents that we know from research that pharmacological interventions really stand to have a huge impact in the quality of life in the long term and in long-term outcomes. And I think that is something that you should take. When you're treating a kid with a neurodevelopmental disability who has like a brain that is a little bit atypical, you really need to be careful, and you might have issues where they just don't respond very well to medications, and you might need to try like 10 different medications before you find the right one. But I do think there's an opportunity for treatment there. And if you start very low and kind of go slowly, you might be able to find a medicine that is able to help your child. And if you are able to help with all of these executive function problems, then their ability to be present for the other therapies is also going to be significantly improved. Finally, I want to say, where are we thinking of from a kind of group? And yes.
I want to leave time for a couple of questions.
Oh, sorry.
We'll be
Yes, so I
at
can.
the end, but why don't you, think about wrapping up the next couple of minutes, and then don't.
Yes, this is my last slide. So we are looking at look at that, and to transition into adulthood and just start looking at post-pubertal kids, because we know that sometimes there are behavioral changes that happen in this group. Obviously, with time, we would love to look at, bring kids over time and do longitudinal studies and natural history. Biomarker discovery is a huge important aspect, because this really will allow us to create targeted interventions, as it was said earlier. Obviously, collaborate with the lab, and you'll hear more about that. And of course, leverage this international collaboration, and you'll hear from Saskia, and of course, you heard from the foundation, that to truly be able to come together. And finally, I just want to help everyone that has been part of this. Of course, this is a multidisciplinary and multi-person effort, and more importantly, to the participants and their families. It's really been wonderful to get to know you and learn so much from you. And I'm really excited that the foundation is up and running. So congratulations for that. Okay, so I'm going to stop. Okay, and that's it.
So maybe what I'll do is pull up a couple of questions. Remember, there's going to be an open panel discussion later. Mm-hmm. Carl Whitney has much better eyes than I do, and looked at your slide with 20 of the mutations, and there counted only 20, and you talked about 22.
Oh, yeah, very, very good. There's a couple of kids that had deletions, right, and that are not included there.
And I think the other thing to note is that Pilar's paper has been accepted, and all the data she has presented will be available in Molecular Autism, which is an online open access journal. And so hopefully in a matter of weeks, all the results will be summarized there together with a narrative. I will say that several people are asking questions about anxiety, about ADHD. How does anxiety manifest itself, and what's the earliest to start ADHD treatment? Maybe those two quick ones, and deal with others later.
Okay, so anxiety in kids can be very tricky. So what I tell parents many times is to think about yourselves when you're anxious. Let's say you're running late to a place, and then a car stops in front of you, and you're like, oh my God, and you start to get irritable, and then you kind of lose your temper. Or maybe not lose it, hopefully, but that kind of irritability, that immediate reaction that you have, and that we've all felt when we are running into a situation that we cannot control, that is what we see a lot in kids with anxiety. irritability is kind of a key component, especially when they cannot verbalize what's happening in them. Of course, there can be other things like having a hard time separating from their parents, having a hard time falling asleep, or really requiring someone to be with them. But I think, a lot of those things, I think intuitively, parents are able to pick up as anxiety. Irritability is one that sometimes is difficult to capture as a parent, because you do not understand why your child would just lose their temper out of nowhere for kind of nothing. And the thing is that probably something is bothering them. So it's like, it creates like a little bomb. And again, like always think about how do you get yourself when you're trying to get something, and then like everything is against yourself, and how that kind of increases emotions. So that is one. And I hope that kind of answers the question. And if not, in the panel, we can talk more about it. In terms of ADHD treatment, that's also a very good question. So there's studies in ADHD that go as young as like 4 or 5. I think the response is less clear. We know that in that age group, what we see the most improvement is like hyperactivity, but not necessarily attention. I generally tell parents that, once a kid is like 6 or 7, we start to see a more clear response in treatment. And of course, it depends on the severity of the symptoms. Another thing about ADHD, especially when you have kids that have kind of very atypical and complex brains, is that you might try a medicine for ADHD at age 5 and they do poorly. That doesn't mean that trying again at age 6 or 7 or 8 is not important, because these brains are continuing to develop, and it could just be that they are not ready at the time you try it. So if you tried a couple of medicines when they were 5 or 6 and it didn't work, I would encourage you to maybe go back at 7 and try again. These are things that you need to constantly be reevaluating.
Thank you, Pilar. And thank you
You're
for
welcome.
this critical work. I think it is important to map the phenotype and understand, as you just pointed out, that some critical features may be undertreated and underdiagnosed, and it's an opportunity to make things a lot better in terms of quality of life, and even in terms of other aspects of the syndrome. So the next speaker is Nan Yang, who is an assistant professor here at Mount Sinai in the Department of Neuroscience. And she's a neurodevelopmental and stem cell biologist. And her work focuses on applying human-induced pluripotent stem cells and using them to make models of neurodevelopmental disorders, answering fundamental questions, biological questions about neurodevelopmental disorders and neuropsychiatric disorders. I will let her explain what induced pluripotent stem cells are.
Thank you, Joe. Can you see my slides?
Yes.
Great. Thank you for the introduction, and thank you everyone for being here and giving us the opportunity to share our work on FOXP1 syndrome with you. My name is Nan, and I'm a faculty member from neuroscience. Our group used human stem cell-based models to study the development and function of the human brain. Together with Joe's center, we are also building human model systems to investigate the function of several genes associated with autism spectrum disorder, including FOXP1. So in today's talk, I will first give you a brief introduction to human-induced pluripotent stem cells, or iPS cells, including what they are, why we use them to study human psychiatric disorders, and how we use them. I will then talk about some of the current work from our group that uses iPS cells derived from patients with FOXP1 mutations and their unaffected family members. So to understand what iPS cells are, we have to take a slight detour and go back to the developing human embryo. Human embryo reaches the blastocyst stage 4 or 5 days after fertilization, at which time they consist of 50 to 150 cells that can grow into different types of cells in our body, including the brain cells, germ cells, blood cells, and other cell types in the internal organ. And their potential to generate all other cell types of different germ layers is described as pluripotency. In 1998, James Thomson successfully isolated the inner cell mass from human blastocysts and put them in cell culture. These cells exhibit remarkable capacity of long-term proliferation. At the same time, they can differentiate into the different germ layers, namely the endoderm, mesoderm, and ectoderm that give rise to the brain cells. Their potential to be used in human developmental biology study, drug discovery, and transplantation medicine was immediately recognized. But you can also imagine the difficulty in deriving human ESLs, both ethically and technically. Therefore, it is probably not surprising that I tell you one of the ESL lines, H1, we use in the lab was derived in the very first study. Actually, we're looking at its image right now. So wouldn't it be great if we could generate something similar to the human ESLs that can be maintained in culture almost infinitely, but still have the ability to differentiate into different types of cells? And this attempt was first performed in other species. In 1962, scientist John Gurdon removed the nucleus of a fertilized egg cell from a frog and replaced it with the nucleus of a cell taken from a tadpole's intestine. This modified egg grew into a new frog, as you can see many of them here, proving that the mature cell can return to an immature state and form all types of cells. However, do we really need everything from a fertilized egg to reprogram the nucleus of a mature cell back to its pluripotency? The answer is no. In 2000.
Okay. I thought you were showing pictures of frogs. I see the frogs. I thought you, yes, I see the frogs now. I thought that was.
Frogs are here, not this one. Yeah, so in 2006, Shinya Yamanaka successfully identified 4 genes. When activated, can turn mouse fibroblasts or skin cells to immature stem cells, which in turn can grow into different types of cells in the body. And this marks the birth of induced pluripotent stem cells or iPS cells. And because of this work, these 2 scientists shared the Nobel Prize in Physiology or Medicine in 2012. So we are very excited about iPS cell technology because now we can derive primary cells from patients, for example, skin cells or blood cells, reprogram them into iPS cells, and then differentiate the cells in theory to any types of cells of your interest for research work and therapeutic development. But why is it important to have these human cellular model systems? What are their advantages over other model organisms? For example, what are the advantages over the broadly used mouse model? Well, first, because we have very different brains, not just in the size. This is the size of a mouse brain, and this is how big our brains are proportionally. But when we scale up the mouse brain, you can see that the mouse brain has a very smooth surface. So these characteristic folds in the human brain greatly expand the surface of our brain. So underlying this structural difference includes changes in cell composition, proliferation, migration, and differentiation. Therefore, many of the cellular features unique to human brain cannot be studied in mice. In addition, there are genes that play very different roles in mouse and human. For example, a mouse bone gene, osteocrine, was found to be expressed in the human brain, but not in the mouse brain. And its expression is regulated by neuronal activity. And people think this has to, it plays a role in our cognition. Obviously, we cannot learn anything about this gene in the mouse brain because it is not even expressed. But to me, what is more important is humans and mice carry very different genetic information. For example, many disease-associated genetic changes cannot be identified in mice. Additionally, we know that different people carrying the same mutation can have very different disease manifestations. And this is at least partially due to the genetic modifiers found in individuals that will be even more difficult, if not impossible, to model in the mice. So the next question is, we have iPS cells, then what are the cell types or cellular processes we want to create using human iPS cells to study psychological disorders? So the large-scale genetic studies led by Joe, among the others, give us some guidance. They found that the expression of autism risk genes, including FOXP1, is enriched in the inhibitory and excitatory neuronal lineages. So what does this mean? Neurons in our brain, they don't work independently. Instead, they form networks. A major way for neurons to talk to each other is by secreting neurotransmitters. Excitatory neurons secrete something called glutamate that can make other neurons more likely to be active. In contrast, the inhibitory neurons uses this neurotransmitter GABA to make the other cells that make connections with them to be less likely to become active. And excitation and inhibition are the two major modes in the brain circuits. When this balance between excitation and inhibition is disrupted, many diseased conditions may occur. Now we have the cell types in our mind. What about the cellular processes? Again, the genetic studies point us in the right direction, which is the synaptic transmission. You may ask, what is that? As I mentioned in the previous slide, neurons communicate. Most of the communication is achieved through synaptic transmission. And the structure that neurons use to talk to each other is called synapses. Humans have a lot of synapses, which are these small gaps at the end of the neurons. So when the electric pulses arrive at the terminal of the signal sending neuron, they can trigger the neuron transmitter release. The detection of neuron transmitter by the receptors on the membrane of the signal receiving neurons will likely trigger some downstream signaling pathways, which marks the completion of the synaptic transmission. Depending on the type of neurotransmitter released, the synapses can be grouped into excitatory synapses and inhibitory synapses. Synaptic transmission responses are regulated by many parameters, and these parameters are complex but can be luckily measured experimentally to achieve the characterization of synaptic abnormality. In addition to active synaptic transmission that is needed, it is also essential to develop a method with high reproducibility, because our goal is not to study one gene or one iPS line from the patient. We want to be able to generate the same cell types using large numbers of genetically distinctive iPS cell lines. And we also want to be able to control the ratio of the excitatory and inhibitory components in our culture system so that we can study if different mutations can change the excitation and inhibition balance. How do we generate these neuron types from human iPS cells? One approach is to trying to reproduce the cell fate changes during development by first turning iPS cells into something called progenitors. They are not neurons yet, and they can also divide. And then these cells are allowed to spontaneously mature into different types of neurons. This approach can recapitulate some critical events during development, but it usually takes months before we can get neurons. Also, the neurons generated in this method are mixed in terms of their cell type identity and maturation stage. Moreover, we have zero control over the proportion of different types of cells in the culture, which also makes it very difficult to reproduce and compare the results. So to address this challenge or these limitations, we set out to find a way to directly convert the human iPS cells into a homogeneous population of neurons. And this is what we found. We found that when one single gene, neurogenin 2, is activated in the human iPS cells, we can get a pure population of excitatory neurons. And when we use two factors, or when we turn on two genes, ACL1 and DX2, instead, we can get a rather pure population of inhibitory neurons. So what about the synaptic transmission that I spent so much effort talking about? We performed patch clamp, a technique in electrophysiology used to study the ionic currents in individual cells to test if neurons have active synaptic transmission. And we do all of that in the signal receiving cells. So as we expected, we can detect currents in the signal receiving cells indicated by these tiny little spikes in the trace. We also use drugs that block either excitatory or the inhibitory neuron transmitter reporter activity to confirm that we indeed have either pure excitatory or pure inhibitory synapses in our culture. So with this method, we can generate in large quantity pure populations of excitatory neurons and inhibitory neurons. And what is more important, we find this method to be really robust among different cell lines. So now we have the platform. I want to move on to some of the work led by a former postdoc, Sophia, who was mentored by Joseph and I. And the data analysis part of this work was assisted by another postdoc, Rebecca Pollock. Sophia had initially three major goals. And then the first one is to characterize the synaptic phenotypes associated with different FOXP1 mutations in human neurons. And then she wanted to know if any of the phenotypes can be explained by gene expression differences caused by FOXP1 mutation. And to do this, she has generated gene expression data and also performed sequencing experiments to assess the chromatin landscapes changes so that she can understand gene expression regulation. But her ultimate goal is, of course, trying to find if there is any pharmacological manipulation that can be used to rescue any of the molecular or synaptic phenotypes that she can potentially find using the FOXP1 human neurons. And today I will brief you with some of the progress that she made. So this is the same slide that Pilar has showed in her talk. I just want to highlight a few mutations that we have already acquired iPS lines and are actively working on them. And this include 2 point mutations that changes 1 amino acid in the protein coding region and 3 frame and shift mutations that likely are creating truncated proteins. I mentioned earlier that synaptic transmission could be measured experimentally to uncover functional abnormality. The most sensitive and quantitative approach is patch clamp, which give you, again, the traces with different spikes, which means currents that you recorded from the cells. And this is just to give you an idea what we usually do. You have to make this electrode and put them to individual cells and record from them. And these are the traces that you get. And these two traces shows what we usually get from a control neuron. And then this one basically shows when we inhibit one of the genes, which is not FOXP1, but another synaptic gene expression, we can see reduced numbers, which we call the frequencies of the spike. That is usually caused by the changes of the numbers of receptor in the signal receiving cells. So this tool is definitely powerful, but every time that we record a cell, we have to get a new electrode connected to an amplifier to record the currents and replace the electrode before moving on to the next one. Therefore, it is definitely very low throughput. And before diving into this procedure, Sophia decided to use another technology, the multi-electrode array, to assay the neurons. So what she used is a plate that have 48 wells. A multi-electrode array is embedded in the bottom of each well. When she plated the neurons in the well, the activity across an entire population of cells could be detected. This method cannot replace patch clamp, but can provide evidence if the neuron property or synaptic transmission is impaired in the mutant cells. And one biggest advantage of this method is you can keep recording for many, many days or even months without damaging the membrane of the neurons. And this slide shows the raw data that Sophia recorded using these plates. Each square here represents the response recorded from each square here from one well, with each of each tick representing one spike, which means if the cells are active. And the recording lasted 100 seconds. This illustrates the degree and pattern of activity of the neurons. And this one is recorded from a control, from the control cells, and then this one from the cells that carry one of the FOXP1 mutations. It is evident that neurons in this well had much more spikes compared to the control well, indicating they were more active. And these spikes here are called cumulative trace. And they show the population spike time histogram, indicating the synchronized activity between the different electrodes. And just again, to show you how it looks, we have multiple electrodes recording at the same time. So if there is network activity and if the neurons manage to synchronize together, then you see all those spikes that are meaning that the neurons are firing together. And we see something very similar with a different mutation when compared to its control. Many parameters.
You're 5 more minutes to the end of the.
Oh, sorry.
Very pretty, thank you.
Yeah, just. This is another slide actually of the data that I really wanted to show to everyone. So we can extract many parameters from MEA, but what we found to be most indicative is the mean firing rate. It can be affected by the neuron's intrinsic property, such as the ion channels, cell sizes, but also it reflects if there's any potential defects in the network activity. And this is what it was from one recording of a pair of control cells and cells with mutation. And the green bars are the data for the mutant. And as you can see, we detected higher mean firing rate in the mutant cells. And we saw something similar, but not as clear a trend in another pair of mutation versus control. We also make sure that this phenotype, or what we saw here, is not because the cells are not firing at the similar scale, which means the mutant is not firing, or the mutant cells fire more, or that's more cells from the mutant phenotype that are active, we don't see that, because you see here, the number of active electrodes are very similar, meaning the activity of both control cells and mutant cells are very comparable. It is the same for this other pair of mutation. So this brings, one thing I want to point out is we always see much higher variability when we look at the mutant cell lines. And this will bring me to our next step. So we want to first perform more MEA recording to increase the number of pairs that we work with, so we can have a better or having a more convincing result. We also, like the experiments we've done so far, use only excitatory neurons. And we also want to add our inhibitory neurons so that we can study the excitation inhibition balance. At the same time, we're working on the data analysis that has already been generated for our gene expression and regulation data. And now we do see something that's different using MEA. We want to perform patch clamp experiments to really understand what causes all the changes that we're observing in MEA. And again, last one is we want to see if we can find any pharmacological or genetic manipulation that can reverse the phenotypes that we observed. So with that, this is the next slide. And we've been working together with Joe's group and generating different human models. It was really fun to work together, and also very important for us to really understand what are the causes associated with different patient mutations. So hopefully we can contribute to the whole study to get a better understanding of the disease and search for therapeutics. Okay, with that, I'm going to stop sharing.
Thank you, Nan. And we're open for questions now. We have some questions about treatment, because obviously parents are very interested in that, and maybe we'll deal with that a little bit at the final Q&A, but we're obviously not in a position to give specific recommendations via Zoom. But I think one of the questions, implied in one of the questions we have here, and also Carl was asking about hotspots for mutation in the gene. Well, maybe the related question is that you might be able to address is there any evidence that there are differences, different types of mutations with potentially different severities?
So it's obviously very important, and that's something we always try to answer. If we can group the mutations into different phenotypes, or just make this genotype phenotype connection. So one experiment that I did not show here was we tried to generate expression vectors with FOXP1 that carries different mutations, and just look at their subcellular location, because that's one of the easiest way to tell how they may work. If they go to different parts of the cell, either the nucleus or stays in the cytoplasm, or even if they change the behavior of their other partner on the cells. So that we do see differences. And even with our MEA, we do see a trend. For example, one of the missense mutations seems to have the highest or the strongest phenotype when the cells are most mature. But then we also see with another mutation, it seems that we have the phenotype shows up, but then somehow disappear after the neurons are fully matured, suggesting that there might be some phenotypes that are temporary, even during development or maturation of the neurons. So were there other hotspots? We don't know. We do have a few that are recurrent mutations that we are extremely interested.
Yeah, so I think that's a really important point, the first one you made, that it seems to be the case that even within FOXP1 mutations, there are types that might have different constellations of overlapping phenotypes, and maybe even some things more severe or less severe. And so one of the tasks that the field has to deal with, and the Family Foundation is no doubt grappling with, is how do we begin to map not only FOXP1 syndrome, but whether there are subtypes of the syndrome to specific mutations, and how does that impact how we might go about treatment and treatment development. So one of the questions, and I think this is also an interesting one, is, well, it's about stem cell treatment. So as you know, there's a lot of talk in the universe, but also even in autism about stem cell therapeutics. And maybe it's worth talking about the differences between stem cell therapeutics and stem cell research and how they, there's what each one's good for.
Excuse me. So the research is important, as I alluded in my presentation, but we also think a lot about if it's possible to develop cell replacement therapy for many psychiatric disorders, including ASD. I wouldn't completely exclude the possibility because there are studies showing, at least using mouse brain, even in adult mouse, if you transplant certain types of interneurons, you can reopen the plasticity. You can change the plasticity. So they can, the mice can learn some tricks that they cannot do anymore because the brain is too old or too mature. But you can just kind of, reopen the key developmental stage. So is that something can be used to correct some behavior phenotypes in animal models and have potential to also be able to help patients? I think there's a possibility. It's just we're not there yet because we don't even know what are the neuronal types that's really affected in different patients that carrying, have different genetic cause of autism. We have to definitely understand that better first.
Yes, thank you. And I'm going to restate what you said, that in terms of stem cell therapy for brain disorders, we are very far away. Maybe not for Parkinson's, but for kind of developmental disorders. And I think one of the things that I think you hear in the press about stem cell therapy, which is where you take cells and put them back in. And we're using stem cells, or you're using stem cells to understand what the brain's doing differently when there's a mutation in it. So, using the stem cells in a dish, it gives a chance to, as you'll hear, as we'll talk about in the next slide, maybe even come up with new treatments. But those treatments would be not necessarily putting the stem cell back. It would be thinking about traditional drugs. Yeah, and obviously there's a lot of work on stem cell therapeutics for the brain, but it's, I think, still a ways away. So I think that's the last question we can handle right now. Nan, thank you for a terrific talk. And I'm very pleased to introduce Anna Kostic, who's an associate professor in the Department of Psychiatry at Mount Sinai. She's the director of the Drug Discovery and Development Program here at the Seaver Center. She comes to us, she's a clinical scientist with expertise in drug discovery and development. She came from industry, and her current research focuses on identifying potential treatments for autism and autism-associated disorders and these rare disorders we're talking about, including designing the screens and assays to find new drugs, and then validating biomarkers to be used as endpoints in clinical trials. So Anna, take it away.
Thank you for a very kind introduction, Joe. And should I share my screen, right?
Yes, please.
There we go. It is a real pleasure and honor to be here today and to talk to this diverse audience. Obviously, the center is focused on FOXP1 as one of the major indications that we're interested in. And I would just like to take perspective on this disorder.
Anna, could you change it to full screen mode?
Yes, absolutely. As from a perspective of drug discovery and development in general, and then I would like to give you an overview of technology that we have implemented in an attempt to develop a screening system for potential drug candidates in organoids, or so-called brains in a dish. So in terms of drug discovery and development, autism is a very challenging area. And I just have a relatively simple schematic here indicating all the different aspects of autism and all the different clinical presentations that could be observed in individuals with autism. This obviously makes the design of clinical studies very difficult because it requires substantial patient stratification, and then extrapolating from one population to the other can be extremely difficult. This is further complicated by the fact that the etiology is not always clear. And this is part of the reason why we have focused on the monogenic forms of autism as sort of a proof of mechanism, proof of concept for drug discovery and development in autism at large. And with that, we're hoping to take a slightly different approach in drug discovery in autism and look at autism as a rare disease. It's basically a constellation of rare diseases, each of which can be explained by slightly different genetic background and genetic variants that contribute to autistic features and autistic behavior and lead to the observed core symptoms. And I couldn't resist the urge to connect these two cute animals and show the rare disease zebra that has tremendously helped the rare disease community over the past 20 years or so, and really paved the way for regulatory approval of a number of treatments in various rare diseases. And I hope, as the rest of the community does, that this approach will continue in many more rare disorders, including FOXP1 and your cute little Foxy that the foundation has just piloted as a logo. So looking at autism as a rare disease has many advantages. First of all, it reduces heterogeneity of the population that you're looking at because all of the individuals have similar genetic features. Understanding these genetic features as well as the functional role of the proteins that are changed in specific variants, it definitely is expected to facilitate the drug discovery and design, both in terms of having a more uniform screening field, as well as in our ability to develop more targeted treatments. And as I mentioned, the rare disease community has been extremely efficient in negotiating with regulators the best path forward for each individual rare disorder and really circumventing some of the paradigms of canonical regulatory approval paths. So we expect that the regulatory path to the regulatory approval for any molecules in monogenic autism and in FOXP1 syndrome per se will be facilitated by this approach. I will not go into the details of this slide as Dr. Boxbaum has already reviewed it. I will just focus on the circled portion, which is human neurons. So we have finally arrived to the place of being able to study brain disorders in the actual tissue that's affected, which is human neuronal cells or neurons. And as Nan so eloquently explained, this ability comes from discoveries in the space of induced pluripotent stem cells and our much refined toolkit that we can use to study these cells in vitro or in the dish. So just looking at these two images, I think is powerful in the sense that it gives us an overview of the complexity of the problem. And as Pilar pointed out, our behaviors are tremendously complex. Our cognitive and executive functions depend on very complex brain structures that you can see clearly from the image on the left are three-dimensional. When we study cells, including human neurons derived from iPSCs, traditionally, we have been doing that in a two-dimensional system. In the Petri dish, looking at the cells adhering to the tissue plastic or glass and their behavior and interactions between different types of cells. This approach has been tremendously powerful and has generated reams and reams of valuable data. We have learned a lot about the way our neuronal cells function and the way our brains work from this approach. But we have definitely missed out on some of the nuances and complexities that come from organizing various cell types into complex tissues and the overarching organ being brain and central nervous system. So to overcome this approach, the field has worked fervently for many years on developing three-dimensional cell culture systems. And as you can see here, and as most of us are painfully aware, the arc of scientific research is very long. So it took us more than 100 years to appreciate and understand how to nudge these human cells into forming three-dimensional structures outside of the human body and to be able to study that. And only recently, in the last 10 years or so, have we really been able to look at the brains in the dish and understand their organizational and functional features. And of course, as soon as these structures and culturing approaches became available, the field has shifted towards thinking about how we can use them in precision medicine, how we can create screening tools and other tools to fit each drug to each patient, develop the personalized or precision medicine approach. And this is just a very nice schematic showing how utilizing organoids derived from individual patients, one could potentially test the drugs in the cell culture and then use this knowledge gained in the dish to better purpose and prescribe drugs to individuals affected by the disease. We have been very lucky to come in contact with a wonderful partner, a very small company called Rumi Scientific, small but mighty. They came out of some tremendous research at Rockefeller University, and they're based in New York and in the Bay Area, just starting their operation, but already deploying this powerful technology in different areas of research. So what the technology consists of is standardizing the preclinical studies of 3D organoids and using them to potentially screen the compound libraries and drug candidates in the dish. So what's different about these organoids compared to organoids in general is that they're grown on the micropatterned surfaces. So these micropatterned chips are basically like a garden, you know, like having the soil in some places and not in others. So when you plant your cells, they would form these organoids in a very structured, deliberate fashion, and the organoids would all be sort of standardized in size and the way they form and organize into three-dimensional tissues. And the beauty of this work that Rumi and Rockefeller have developed is that they are able to do it in very, very tiny little dishes called 96 well plates, and even tinier ones that are called 384 well plates, which means that you can screen hundreds of conditions in just a single dish. And within each little well, there is hundreds and thousands of these little organoids. So you really ramp up your numbers and gain the statistical power to analyze the data in a clinically meaningful way. What's also nice about these organoids is that through the synthetic reconstitution of iPSCs, Rumi was able to differentiate organoids that contain different cell populations that are relevant in the brain development. They're also capable to grow them to different stages of development that very much resemble the development of human brain during fetal development. And how does this fit into a much bigger picture? So we're hoping to really close this circle of drug discovery and clinical research and start with our knowledge of genetics in individuals with autism, look at patient-specific mutations and genetic variants that we believe give rise to certain behaviors and phenotypes, then generate out of patient cells organoids that would represent the tissues and behavior and phenotypes that we think are critical for the disorder. We would use machine learning for the analysis, so it would be very unbiased and not really dependent on human perception and subjectivity, but it would be all done through automated algorithms that Rumi has very carefully developed. And then once we have the drug candidates put through this discovery pipeline, we would proceed with the drug validation in vivo in numerous mouse models that we have. And Jenna will be speaking later about amazing work that her group is doing in the FOXP1 mouse. And after validating the potential drug candidates in these preclinical systems, we would go into the clinical trials with a lot of information in hand and really be able to design these trials in the most informative way and test the drugs with the greatest chance of success. And I'm very, very excited to show some of the preliminary data. And I want to thank Rumi again for giving us the permission to share this with you as a community and demonstrate that we are actually capable of using iPSC lines with FOXP1 mutations on this platform and generating what we believe are strong enough signatures that would allow us to screen drug candidates and select the ones with the potential of reverting the mutant phenotype back to what we see in healthy, quote unquote, controls. So this is the first organoid that Rumi has developed called neuraloids. And without going into this very detailed and dense data slide in great detail, I know we have a lot of non-scientists in the audience. I'll just draw your attention to a couple of features. So if you look at the parental line, this is the line that does not contain any of the pathogenic FOXP1 variants. So you can see the organization of the organoid, and all these different colors represent different markers of the cell development. And this is what you would expect the neuraloid to look like at this stage. If you look at the FOXP1 heterozygous cell line that we have developed through genetic engineering on the same parental background, by introducing just one mutation, and then you look at the homozygous cell line with both copies of the mutant FOXP1, you can see that the structure of the organoid is tremendously changed and that the distribution of the markers and cells is deeply altered. When this is measured through the morphometric measurements that Rumi has developed and validated through a number of optimization studies and protocols, you can see that there are deficits in neural and neural crest induction, and the classification of this phenotype and validation was successful to the level that we feel that this is consistent and reproducible. We can replicate this data in multiple organoids in multiple dishes, and we hope that this could present the basis for further screening. And with this, I will take you to the second organoid that we have looked at, and this is so-called micropattern neuroepithelium. So this is just slightly later in the development. And again, the similar theme emerges, and you can see how the parental organoid looks very, very different from FOXP1 mutant organoids. And you can see the in-depth analysis showing a really good separation and statistically significant difference between different genotypes. So this is exactly what a scientist would look for in an assay that would allow us to measure the drug effects. Because if the differences between these genotypes are smaller, it would be very, very difficult to evaluate the effect of the drug. But once you have a large gap, it's much easier to see if the drug treatment would close this gap. And the final organoid that we looked at is called early cerebroid. And again, we see differences, and perhaps they're not quite as pronounced, but we feel that there is still a pretty strong phenotype that could be, again, accelerated into the screening campaign. And with this, I will lay out some of the next steps that we're planning. As exciting as these data are, they're just the tip of an iceberg, and we would like to confirm these findings, these early preliminary findings, in more cell lines. So all the work that you have seen was done in genetically engineered lines. We would like now to take a step further into the patient-derived lines. And for this, we are incredibly grateful to, first of all, the individuals affected by FOXP1 syndrome for donating these samples to science, and also to their families who have not only donated samples themselves as sibling or parent controls, but who have also come to the Seaver Center and took part in our research and the assessments of this relatively still novel disorder. We currently have 6 patient-derived cell lines, and we plan on proceeding with analysis on Rumi's platform to better understand these observed phenotypes and to hopefully be able to move this research forward towards testing some drug candidates. We will select different compound libraries out of the existing libraries, and when I say that, I mean we can look at the currently approved drugs for different psychiatric disorders or neurological disorders. Pilar spoke at length about the value of controlling and managing the aggression or attention deficit and hyperactivity disorder within the context of FOXP1 syndrome. We could focus on the drugs that are already approved in these disorders and test them on FOXP1 deficient cells. We could look at other drugs that are capable of penetrating blood-brain barrier, and we can also conduct completely agnostic screens of large number of molecules that would then later on be subject to medicinal chemistry and further chemical modification to really bring them to the level of a drug. And then finally, we can use this powerful platform to evaluate some of the targeted drug approaches that would go strictly after the modulating FOXP1 expression or correcting some of the downstream signaling. Once we perform these screening campaigns, we are hoping to identify some potential hits and then further validate them in secondary assays, including the mouse models, and other in vitro models, such as the ones that Nan was describing in her presentation. And with all of this collective effort, take us a step closer to having treatments available to FOXP1 syndrome individuals, as well as individuals with autism that's not quite genetically elucidated yet, and that we currently think of as idiopathic. And with this, I would like to thank Beatrice and Samuel Seaver Foundation for their unwavering and generous support over the last almost 30 years. I would like to thank Dr. Boxbaum for his mentorship and support. All of the Seaver Autism Center, it's a wonderful group of people. We love working together, and we feel privileged to be working with our patients and their families who have shown tremendous support for our research as well. And I hope that all of us together will be able to really move the needle and bring some novel therapeutics to the field of neurodevelopmental disorders. Thank you.
Thank you, Anna. So I have time for a couple of questions. One of them is, what kinds of cells and how many cells are in the organoid? When some of the other FOXP1 associated genes like FOXP2, has anybody looked at the same organoids with related genes and seen something similar or different?
So organoids are incredibly complex, and we're talking about the thousands and thousands of cells, probably even millions, I think. Again, unfortunately, I don't have Fred, Itak, and Ilona Nemeth from Rumi Scientific here on the podium with me, but I'm sure they're in the audience, and they could give you much more detail on their technology. I'm unaware of other FOXP genes being used on this platform, but Rumi themselves have studied other lines, not necessarily in neurodevelopment and autism, but in neurodegenerative diseases, and they were successful in demonstrating very strong phenotypes as well as reversal of these phenotypes with potential drug candidates. And the Seaver Center has actually shared additional cell lines from other disorders that we're focused on, including ADMP and ADMP syndrome and DDX3X syndrome. And we are happy to report that phenotypes were identified in these lines as well. We are currently in the process of expanding this research in all 3 genes to look at additional patient-derived lines. And in the meantime, Rumi has perfected yet another organoid later in development called late cerebroid. This organoid is better suited for another gene close to our heart, which is SHANK3. So we will be expanding our research and collaboration in that direction as well. So I am hoping that a year from now, I will be back here presenting on a little matrix of organoids with different genetic deficits and different phenotypes, and hopefully some screening results as well.
Be great. So there are a couple of questions that I'm going to link together. One family is asking about if they had banked cord blood. Would that be useful? Could that be beneficial in any way? And then a related question we're getting a question about tissue or DNA from somebody who's 20 years old. Would that be useful in some of the work that's going on?
I cannot emphasize the value of having access to human tissue in research in general and drug discovery in particular. It's always good to have samples from actually affected individuals, because only in the context of these samples can we really study the true complexity of the interactions between the genotype and phenotype and really understand the molecular mechanisms behind it. In terms of the cord blood, thanks to the technology that we're describing here for iPSCs, we don't necessarily need to tap into your cord blood supply, save that for, God forbid, more difficult circumstances. But we can very easily derive cells from any individual with just a simple blood donation. We have moved away from skin donations that are more invasive. With a simple vial of blood, we can generate lines of iPSCs and subsequently neurons. So that is our preferred approach at the moment. And then I will let you, Joe, address the question of the DNA, since you have led the fantastic initiative that Autism Sequencing Consortium have conducted. I think every sample counts. So having additional samples is always welcome in terms of studying the genetics. And accompanying blood samples are always good as well.
Yes, so I agree that the samples are great. And I wonder, Carl, I don't mean to put you on the spot, but I'm sure that there's at least been some discussion or some thinking about discussing, banking through the foundation. And I'm sure you're not ready yet. But is that you may not even want to talk about it, but if there's something you want to add.
I'm happy to say that. I mean, we have definitely talked about it. We don't yet have a solution in place for that, and that's definitely an important consideration. Having those biobank samples for future questions as technology advances is critical. Maybe it's an area for common collaboration with groups like the Seaver Center for the future.
And we have a couple of questions about FOXP1, first of all about animal models, and second of all about their role in differentiation of nerve cells. And I think maybe Jenna will talk to some of that. So maybe we'll leave that. We can always come back to it in the open question and answer. And one of the questions that we have here, which is a good one, that we seem to mostly be talking about mutations, and are the studies we're talking about also valid for deletions? So Carl asked about the 22 cases in the Seaver Center. We have 2 that are deletions and 20 that were mutations. So it's a fair question.
I'm sorry, I'm an old school biological scientist. So when I first started studying genetics, we had this classification of mutations indicating any variant from what is considered the wild type genome. So that included point mutations, deletions, insertions, all the different types of genetic modifications or variants that you can encounter in the human population. So when I say mutation, I generally just mean a variant that's associated with FOXP1 syndrome. We know that there's a number of variants that are considered non-pathogenic. We all have our little differences at the level of individual nucleotides. And when we talk about mutations, we're just focusing on those specific variants of the FOXP1 gene that have been associated with phenotype. So that includes deletions in short.
But I think it's an important point. We are at the field, and I'm sure the Family Foundation is always paying attention to different kinds of mutations to see if there's any differences. But so far as I know, the deletions and most of the point mutations look exactly the same, both in terms of molecular work, well, in terms of phenotype. I'll stop there. Okay, so thank you.
Thank you.
And our next speaker is Saskia Koene, who is a clinical geneticist in training from Leiden in the Netherlands, and did a PhD at the University of Nijmegen, and trained as a clinical pharmacologist with special interest in the intervention studies of rare diseases. And she's reached out to Pilar and others, and I'm sure many people in Europe as well, to develop a questionnaire, which she'll describe briefly now. Saskia, thank you.
Thank you, Professor Buxbaum, for the opportunity to present to this wonderful audience. What we did was we designed a questionnaire to elucidate what problems children and adults with FOXP1 syndrome face. And as you mentioned, I didn't do this alone. I asked for input from more experienced researchers, for example, Pilar, and also the authors of the paper on the language. And they were very happy to help me with designing the questionnaire. And I also had some help from Annemiek and André and Esther from Kind Beter Foundation. So all that input led to this questionnaire. And the study aims to provide parents and practitioners with more clear information after the diagnosis has been made. So regarding development, regarding what will my child be able to do independently in the future, all that kinds of questions are the questions that we will seek answers to. But of course also to medical questions like, is there a hole in the heart? Was there some medication that has been tried and gave tremendously good effects? That will be the inventory to which we designed this study. What will we ask from you? It is an online study. So the survey can be filled out online. But first we will need informed consent, and we need to verify the diagnosis of FOXP1 syndrome. As has been said, you have deletions and you have mutations. We are looking for pathogenic mutations and children and adults with deletion only, including FOXP1. So no larger deletions, only FOXP1. And if you have a letter of the clinical geneticist, we can read from that in which group your child or the one that you represent is falling. After the diagnosis verification and informed consent, you will be sent a link to the questionnaire. The questionnaire is now available in Dutch and in English, but if many people from France or from, for example, a Spanish-speaking country will ask for the questionnaire in that language, we will be able to translate the question. That also might take a while, but we eventually will manage. And then there's also a possibility to upload some medical reports. You'll have to answer a lot of questions, and then there will be at the bottom a possibility to upload some reports if you're not sure about if you filled it out correctly, because there's quite some medical terms in there, unfortunately. And if you have sent the questionnaire back to us, we will put the data in a safe box. So it's coded, not traceable to your child, but the informed consent letter states a lot about those kind of details. And you will get sent a summary of the report and also the scientific report at the end of the study, and maybe some advices on management that will flow out of our study. This study doesn't stand alone. In the LUMC, we have a research line in FOXP1, also elucidating the immune system. But this slide depicts the study of Dr. Willeke van Roon and Elena Dazzali. And they are also trying to make FOXP1 brain organoids with a specific mutation. And their final aim, together with the Kind Beter Stichting, is to design a DNA repair therapy. And Willeke is also available, so if you have any questions, she might also be able to answer them. And this is my summary slide with all the details on the study. And if you want more information, or if you want to be included in the study, please contact me at foxp1@lumc.nl. And for now, I'm also very happy to answer any questions.
Thank you, Saskia, and thank you for putting together this important survey. Questions for Saskia? I think it's very important that the international community is all kind of working together, because for rare disorders, you know, it's the only way we can make progress. So one of the questions we have here is, how are you deploying the survey?
Sorry, I didn't understand correctly.
How do you deploy the survey? How do people sign up? Is it web-based?
Oh, yeah, it's a web-based survey, and you can send an email to foxp1@lumc.nl, and then I will give you more information about the study and how to be part of it and what it includes to be part of the study. And then I need a letter from the clinical geneticist, or just a genetic result. And then if I have those details and I have assigned informed consent, then I will send you a link to the questionnaire. And the questionnaire is in Castor. That's a super safe system that we use in the LUMC all the time for patient data.
Well, a lot of questions about, is it open to international, like US participants? They're asking for your email address again, which we will send around. Yeah, so what we'll do at the end, we'll send around your contact, the FOXP1 at the email address you sent around. And I'm gonna I'll let you answer this, but is it open to US citizens? Unlike travel to Europe, is the survey open?
The survey is open. You can send an email now, and then I will answer tomorrow because it's quite late here. And then maybe you can have the link before the weekends if we. And
what languages? What languages do you have the survey in?
Dutch and English.
All right. So that should cover many people in the audience.
Probably. And otherwise, you can just send an email, and I can try to find someone to translate. I don't have many financial resources, but there will always be.
I'll translate it to Spanish if that's helpful.
I will also put your email address in the chat for everybody to see, but we'll send it around after this meeting as well to everybody who registered.
Thank you very much. Thank you.
Thank you, Saskia. So our last speaker is Genevieve Konopka, who's an associate professor in the Department of Neuroscience at UT Southwestern Medical Center. She received her PhD in neurobiology from Harvard University. Where is that exactly? I've heard of it. And did her postdoctoral training in neurogenomics at UCLA, which is on par with Harvard in terms of neurogenomics. So two of the best institutions in the business. And she's run a laboratory at UT Southwestern and Department of Neuroscience for the past decade. And she studies the role of genes implicated in autism in brain development and in autism phenotypes. So thank you, Jenna. Looking forward to your talk.
Thanks for the introduction, and it's really my pleasure to be here today. This is a wonderful event. I'm going to start sharing my screen. Let me just click on, and then I usually like to use a laser. All right, can you see that, Joe? Yeah.
Yes.
Great.
Excellent.
Thank you to all of the families for asking all of these wonderful questions, and for those of you who are already participating in research with the Seaver Center and around the world, it's a wonderful community, and I'm really excited to be a part of the new International FOXP1 Foundation. So thanks again to Carl for including me in that. So lots of introductions, but I promise I'm going to get us back on time because I don't want to overwhelm with too much jargon, but I do want to share with you some of our ongoing work, and almost everything I'm going to show you is unpublished. So, and here these pictures are from a mouse brain, pretty pictures from my graduate student Anna. So I don't think I need to really explain what FOXP is to this particular audience, but I did want to re-emphasize the fact that FOXP1 is a transcription factor and what that means. So what that means is it's a protein that basically sits on DNA and turns on and off other genes. So that we think is the major function. And so as a neurogenomics lab, we aim to understand what genes FOXP1 is turning on and off. Essentially. And in this study, we're trying to understand how it does that throughout brain development. In a couple of my first intro slides, I also will show you images of FOXP2, and someone already brought up a question about FOXP family, other FOXP proteins. And what's very interesting about FOXP2 as well as FOXP4 is that there's evidence that they can interact and potentially work together with FOXP1 in the brain. So if you have an individual who has a FOXP1 mutation, it's possible that this is not only interrupting the function of FOXP1, it could be interrupting the function of FOXP2 or FOXP4. On the other hand, these other proteins may be stepping in to replace the function of FOXP1. So it becomes very complicated when you start thinking about how proteins interact with one another. And we know this is in a brain, well, it's in an organ and a brain region and in a cell type specific manner. At least that's what my lab works from that hypothesis. Okay, so on the right-hand side here, what I'm showing you is the expression pattern of both FOXP1 and FOXP2 in the human brain. So this is data taken from brain tissue from individuals who had passed away throughout the lifespan. This dotted line indicates birth. And each one of these colors indicates a particular brain region. So the striatum, the neocortex, the thalamus, the hippocampus, and the cerebellum. And what I'm highlighting here is the expression of FOXP1 in one of those regions, the neocortex, because that's what I'm going to focus on today. And hopefully what you can appreciate is that there's really kind of these two bumps of expression throughout development. I'm going to illustrate this in another way in our data from the mouse, but to us, this suggests that FOXP1 may have one role very early in brain development, and then it may have potentially a different role later on in brain development, in this case in the human, even before birth. And just to contrast that, FOXP2 expression seems relatively even and lower, because the height here of this graph indicates the amount of expression, but I will illustrate in the next slide why that might also be. So here is just an kind of a schematic of a cartoon of a mouse brain, if you will, generic brain, and these colors indicate where either FOXP1 or FOXP2 are expressed. And so hopefully you can appreciate in the neocortex here, this outermost part of the brain, there's really high FOXP1 expression. Also in the striatum, and at the family meeting 2 years ago, I talked about our work on FOXP1 in the striatum, as well as the hippocampus, which is very important for learning and memory. So this is just a zoom in into the neocortex. The neocortex is very well characterized for its what's called its layering or its lamination, starting down here in the subplate and then going from layer 6 all the way up here to layer 1. And again, FOXP1 is high expression throughout, especially in these upper cortical layers, whereas FOXP2 is more greater expression in the lower layers. Why is that important? Well, the cells in each of these layers have different functions. So for example, FOXP1 has high expression in these upper layers, and these cells or neurons are known to project within the neocortex, so talking to one another. They also are expressed in the neurons that project to the striatum. And I just told you, we and others have been doing a lot of work on the role of FOXP1 in the striatum. And in contrast, FOXP2 is in neurons that project to this other brain region, the thalamus. So now I just want to zoom into one cell here that's kind of at the interface of layer 5 and layer 6, where we know there are neurons that express both FOXP1 and FOXP2. And since these proteins are transcription factors and regulating how genes turn on and on, they primarily spend their time within the nucleus of the cell, illustrated here, although they can translocate in and out. And we have other work on what might be modifying that. And work from our lab and the work from a lot of other labs have been really trying to identify what are these downstream genes, and one of the chat questions was exactly about that. And so for FOXP2, there's been a lot of work to suggest it regulates dopaminergic signaling and synaptic function, where our work and others has shown that targets downstream of FOXP1 are indeed those involved in what the person asking the question was getting at. Are they involved in basically turning on neurons? And indeed, we do believe that FOXP1 is very critical for neurogenesis, and also, as you've heard already from Anna, is involved in likely involved in synaptic function and neuronal activity. So it's a bit complicated how these two family members, protein family members, may be regulating similar or distinct target genes. Okay, so now I just want to share with you just an overview of how the cortex develops, because I think that's how we and others are trying to understand the role of FOXP1 in the neocortex, either in the organoid system that you've been hearing about, or iPS-derived models, if they're being pushed towards a cortical fate, and of course in the mouse. So I'm going to be zooming in here again into this region of the brain, the neocortex, and basically where it lines these ventricles here. So this is a very busy slide. It's not my slide, it's taken from a review, but it really, I think, highlights a couple of things. One, the complicated nature of cortical development. What you're seeing from left to right is how this happens from this is in a mouse brain, it's illustrating, from embryonic day 10 all the way to a fully formed postnatal brain. And then all of the different cell types that are involved in building this wonderful structure we call the neocortex. And I hope what you can see down here is that cells can undergo different divisions, and eventually you will start forming neurons. And the way that the neocortex develops is in an inside out fashion. So we have these progenitors down here in the area touching the ventricle. So it's called the ventricular zone, with the subventricular zone right above it and an intermediate zone. And now at at some point around E13 in mouse development, you start to form the very beginnings of this laminated structure of the cortex. So you get layer 6 first, and then cells migrate through layer 6 and form layer 5, and then through into 4 and 2, 3, and so forth. And so it's a very well orchestrated process. And because it's so well orchestrated and has been studied for many decades in the mouse, we can easily dissect this process and study how things go awry when we have certain genetic manipulations, such as modifications to FOXP1. And you already heard from Nan that there are certain cell types and certain aspects of, for example, a mouse brain that are very different than in a human brain. And I totally agree. And my lab also studies human cells and tissues. But I think for these basic well-conserved processes, such as the sequential steps of cortical development, we can indeed use the mouse model. Oh, and layer 1 up there. Okay. So, which brings me to my next slide. And if you were at the meeting 2 years ago, I used this slide to kind of, justify our use of mice. We've been using them for a long time, but they're not a small human. But as I just said, there are some similarities. And so we can use them to study those processes. And importantly, what we can do in a model system such as a mouse versus using our cellular models is we could study behavior. That I'm not going to show you behavior data today, but that is one advantage of a mouse. We also have collaborated with Todd Roberts' lab at UT Southwestern to use a different model, a songbird model, because songbirds have learned vocalizations, whereas mice only have innate vocalizations, kind of like just the babbling of a child. And so we've also manipulated FOXP1 in that model system. So one difference that has already been noted is that mice have these, a smooth neocortex, and humans have these folded neocortex, but the way in which the neocortex connects to other parts of the brain and receives connections is well conserved between species. And also that patterning, excuse me, the lamination is also conserved. Okay, so the data that I will be sharing with you today comes from one of the mouse models that we've been working with. We have numerous FOXP1 relevant mouse models, but this one in particular is what we call a conditional knockout, where we're just targeting, in this case, we're going to completely remove expression of FOXP1 in a specific brain region. In this case, the cortex. It also targets the hippocampus, but we won't be studying it for the purposes of today's talk. So here we just have an animal line that, again, targets using Krelock's technology, if you're familiar with that, targets the neocortex as well as the hippocampus. And we cross it to this mouse that has the FOXP1 gene tagged with LOXP alleles. And then when we breed these mice together, we can either get control mice that have full intact expression of FOXP1, or animals where we've conditionally knocked out FOXP1. And so this is from one of our previously published studies. Green here is highlighting where FOXP1 is expressed, and you can see this beautiful expression throughout the neocortex, as I already described, primarily in the upper layers. You can also see expression in the hippocampus, as well as the striatum. And when you use this genetic targeting method, you see the striatal expression is retained because we're only targeting the neocortex and the hippocampus. But today I'm only going to be showing you developmental data from the neocortex. However, these two previously published studies, which I believe there are links to them on the website of the foundation and some description of some of them. I'm not going to go over those, but I'm happy to talk about those data. I'm just going to summarize the behavioral phenotypes, which again is an advantage of the model. So these animals have decreased sociability. They have impaired spatial learning. We think this is probably primarily because of loss in the hippocampus. They have decreased postnatal, so early born animals, and then adult vocalizations. Again, these are innate, not learned, and they're also hyperactive. It's not advancing. Okay, here we go. So let me go back. Sorry. So this is also taken from one of those studies where we just took a section through the brain and we looked to see in the neocortex what was going on. So first I'd like to point out here is FOXP1 in purple. Again, you see it through primarily these upper layers, completely gone in the knockout on the right. And then we just have different markers for the different layers. So CUX1 for upper layers, CTIP2 for lower, FOXP2 for layer 6, and this DAPI just marks all cells. So the first thing we noticed was that the upper layer, so by using this CUX1 expression, was reduced. And I'm going to summarize this on the next slide. We also interestingly saw in the knockout that there were CUX1 positive cells down here in these lower layers, which is unusual. So this would suggest perhaps cells are migrating up through, as I explained this, inside out development and getting stuck here. So it could be impaired migration, or that these cells are abnormally turning on CUX1 expression. Those were, two, possibilities. In addition, sorry, it didn't advance. Well, here we go. We saw that FOXP2 expression was reduced in the lower layers. Sometimes my PowerPoint gets stuck and it's stuck again. Hopefully it'll advance to the next slide. Let's try it. Oops, see, now it's going back. There we go. All right, this is an important slide. This is summarizing all of this work. So what these studies have shown is that in this genetic mouse model, we have the overall thinner cortex, neocortex. You can see the size is different. We've increased relative upper layer, increase of these thickness here, increase this weird mispositioning of these cells that belong in the top but are stuck down here in the bottom, and then a decreased lower layer thickness. Okay, so together this suggests that there's something going on early in brain development, because here we're looking at the mouse brain just 7 days after birth. So we decided we had to look in an embryonic mouse. And so this is the work of a graduate, very talented graduate student in my lab, Anna Ortiz, and she's asking the questions, when and where is FOXP1 important for cortical development? Which cell types are most affected by loss of FOXP1? Does FOXP1 perform distinct roles in the dividing cells versus neurons? So these proliferating or progenitors that you already heard about. And what are the gene targets that are responsible for each of these functions? So here are some nice images taken from a paper published by Ben Novich. I believe this paper is also listed on the Foundation's website, where hopefully you can appreciate, so if there's dark staining that indicates FOXP1 expression, that FOXP1 is expressed down here very early in these proliferating progenitor cells, and then you see it later on turning on in these neurons that have migrated up and are positioned in the cortical plate. And so this is a schematic from their paper showing what we talk about in the lab as having an hourglass expression in the neocortex. So it's on really high in the progenitors, and then over time it becomes really high in the neurons that are what we call postmitotic. They're neurons. They're not dividing anymore in the neocortex. So again, Anna wanted to focus on these start early and think about these proliferating cells because of the phenotypes that we have previously found in the animals after they have been born. And so just to remind you, this is because, again, of this decreased cortical thickness, we thought there must be some abnormality with proliferation. And so what Anna decided to do was to use a BRDU assay. And basically what this is a chemical that gets into the DNA of a cell, but only when it's dividing. So you can add it at any point, and in this case, she's going to add it very early around E12.5, and we know there are a lot of dividing cells. And then you can wait a certain amount of time, and it's coupled to a fluorescent marker, and you can measure the number of cells that took up this chemical DNA patterning marker, if you will, at this point in time. And so we can do this at different points in development. We can add it in for different amounts of time, and we can couple it with other measurements to understand how proliferation might be affected in these animals. And so I'll just kind of walk you through what this experiment looks like, and then I'll show you a summary of the results. And this is still ongoing work, as it's not done yet, but I always get excited to show the unpublished and things in progress we're doing. So here, again, we're sectioning through the brain. We're just looking at the neocortex, and down here between these lines are really where the proliferating ventricular and subventricular zones are. And then within the thin line, because we're really early in development, you don't really have much of these neurons yet. And that's illustrated here. So SOX2 is marking the proliferating cells, TUJ1 is marking these neurons, DAPI again is marking all the cells, and here's our BRDU label. So you can see it's greatest down here in the proliferating zone. You're starting to see some cells up here. That means that they were marked and then they moved up, and we can still see the mark. And Anna carried out the same thing in a knockout. Now by eye, it's pretty hard to see any major differences, but what Anna, and she had some summer students this past summer working remotely, helping her quantify many of these images from many animals at different time points with different parameters. And I'll just summarize for you some of her results from one time point, that she did find a significant decrease in the number of cells that took up BRDU in this knockout model. And also using SOX2, she just found that in general, the number of these progenitors themselves were reduced. And also the number of neurons in the, the newly formed neocortical plate were also reduced. So we have multiple things going on here. And it could be that the reduction in the number of neurons is because there's a reduction in the number of progenitors and proliferating cells. So those are things that we're trying to tie together. She's also looking at other time points to try and create a model of what is going on with FOXP1 at different time points. So that is just a snapshot of a lot of ongoing work that we're doing with these embryonic mouse brains, and they're very challenging because we have to breed the animals in a certain way to know we're at the exact right time in development. So this is kind of a summary, just illustrating that in a normal brain, you would see quite a number of proliferating progenitor cells here lining up against this ventricle. But when you knock out FOXP1 in the cortex, you have many fewer dividing cells. And we think this is indeed probably one of the major mechanisms for why we're seeing a thinner neocortex. Okay, so what I'm going to spend my last few minutes on is trying to get at the gene target. So again, this is a very complicated process, and you have many different cell types, and we know that the gene expression in each one of these cell types is very different. So if we wanted to know what was going on with the targets of FOXP1 in the brain, and if we just took a chunk of the brain out and then processed it to see what genes are turned on and off, we wouldn't really know did the change occur in the proliferating cells, did the change occur in the neurons, because we're really trying to, again, understand this process of gene regulation by FOXP1. So we've been turning, so here's the overall approach. It's to, let me just make sure I didn't, okay. Couldn't remember the order. She dissects out the cortex. She looks at 3 different time points. So E13, when we know FOXP1 is expressed in the proliferating cells and in the neurons, E16, where it's shifting more into the neurons, and then finally at birth when it's completely in the neurons. And this is taken from previously published work, not from our lab, Chris Walsh. And then she collects at least 4 animals for each time point, either control or knockout. And then she's using this approach of single nuclei RNA sequencing. And I think I should have a slide. Yes, here it is. Okay, so I also have a video, but I can never get it to run with the laser pointer. But anyway, this is a microfluidic system where we're pushing through individual cells into oil droplets. And then we're capturing each individual cell in a droplet, and we're able to measure every RNA molecule, basically, which is the output of what FOXP1 is doing. The DNA turns into RNA, which turns into protein. So we're capturing all these RNAs, which are an indicator of what genes have been turned on or off, and we can quantify them. And then what we end up getting out of this whole process is this massive matrix where we know the expression of every gene, so this is like 10 to 20,000 genes, how they're expressed in every cell that we captured. So it's big data, and then we can visualize the data, do a clustering analysis and say, oh, these red cells have a similar overall expression profile and compared to these blue cells. And then maybe we would say, for example, these would be the progenitor cells and these would be the neurons. And this is just an example of what a real data set would look like. And every dot indicates an individual cell. This is an overview of this process. And I thought I'd have time, but I don't think I really have time to go through it, but it's just to remind you that the brain is very complex. We can isolate individual cells in these droplets. And then through this technology, we can get the cell type specific expression. We can quantify with absolute precision each gene's expression. And in an interesting way, we can also see, say FOXP1 is expressed in this cell, but not this cell. We might see changes in a cell that doesn't normally express FOXP1. And so we'll see what is called this non-cell autonomous change. So how cells can like affect the expression of other cells, or genes in one cell can affect the expression of other genes in different cells, which makes it even more complicated. But now we have the technology to measure all of these things, and we didn't appreciate it before we had this technology. So I didn't want to bore you with a lot of these plots, but I'm just going to highlight one example data set from E13, and where she took the whole brain, and we did there, I think in general, there are like 60 or 70,000 cells that we processed, but she just wanted to highlight the cells that are involved in this neuronal, excitatory neuronal lineage. And so we had about almost 10,000 in the knockout and more 20,000 in the control. I don't think that is a biological, that's just more of a capture technological reason. But nicely, what we saw was that the profile, as we might expect, clustered based on what we think are those cell types. So here are all of these indicate cells from the ventricular zone. All of these dots indicate cells from the subventricular zone. And then all of these are the deep layer neurons. And then we know whether it came from a knockout or a control, and then we can examine what genes are changing in a cell type specific manner. And another cool thing that we can do with this data, and I wanted to use this even though it's a bit tricky to understand, even for people in the world of neurogenomics, we can generate what's called a pseudotime. And so we can take all of these cells that we captured at E13, and we know a lot of stuff's going on, right? Cells are proliferating, cells are migrating, cells are dividing. And so they're all in this different kind of time frame, if you will. But we can use statistics to infer the order of which cells are at which stage, for example, of differentiation in the neocortex. And here's what a plot of that data might look like from the control animal. So here we can see this histogram where the gene expression profile is coming from the ventricular zone, and then from the subventricular zone, and then from these deep layer neurons. And when we make the same plot from the knockout animal data, I hope what you can appreciate is there's this massive shift to the right, and also the three kind of profiles clumped together. So what this is saying is there's a massive delay in development in the knockout. And we see this for each type of cell. And what we can do now is drill into the changes in expression in each cell type and try and understand what are the target genes that are making this shift. We can take those lists, we can put them into analyses called gene ontology to identify what's going on from a biological process. And not unexpectedly, we see genes that are involved in neuronal differentiation and neurogenesis, neurodevelopment. So all the things we might expect. But now, I didn't list them here, we do have those specific gene targets. So for example, Anna was telling me about one transcription factor that she's identified downstream of this already transcription factor that she thinks is important for differentiation, SOX5. So we have something to go on. So those are in theory, these druggable targets that you've been hearing about. So I'm just going to summarize all of her work in my last minute from these three different time points, just to give you a flavor of what we think is going on, and it's still ongoing. So again, at this early time point, we think that the proliferating zones are the most impacted. And again, we're seeing these genes involved in cell proliferation, also migration, and these are being decreased when we knock out FOXP1. At this middle part of development, E16, we're seeing that newly committed neurons are the ones that are most affected by FOXP1, and we're seeing genes involved in kind of the genes that kind of turn on neuronal differentiation affected. Also, genes for cell proliferation continue to be affected and are decreased. And then at birth, we're seeing a reduction of these overall just capturing deep layer neurons, which fits with our previously published work. And when we overlap these gene sets with known genes, and a lot of this comes out of the work that Joe's group is doing, these autism gene lists, we get a significant overlap, suggesting that FOXP1 is regulating other genes implicated in neurodevelopmental disorders, in particular autism. So with that, I'm going to end. I'm going to thank my lab again and my funding agencies. And most importantly, I just want to again thank all of the families for their participation and for learning along with this. And I'm happy to take questions if there's time.
Plenty of time. Thank you for a great talk. And I said Harvard was great and UCLA was great, but UT Southwestern is also one of the leading sites in the world for neuroscience and psychiatry. So I didn't want to miss that point.
Oh, no.
We do have a couple of questions that maybe you'd like to tackle. Here's a subtle one or a specialist one, and maybe you can explain why the question's raised and how you answer it. So have you done any parallel studies using FOXP1 heterozygous animals?
Yes, that was our very first, the first paper the lab published actually. And so we used the heterozygous map. We put the heterozygous animal through a number of behavioral, sorry.
Maybe you can first explain what's the basis of the question, heterozygote versus homozygote and those sorts of questions.
Oh, I'm sorry. Yes, I missed you reminding me to do that. So in this case, we completely removed, so we have two copies, right? And we deleted one copy of FOXP1. So there was still one copy remaining. And when we looked at how that translated into how that loss at the DNA level translated into expression of FOXP1, there was about a 50% reduction of FOXP1 expression in the brains of these mice. But it was only in certain parts of the brain, which was really interesting. So in the neocortex, which I just talked about for the last 30 minutes, with the loss of one copy of FOXP1, there was no effect. So to me, that suggested that it's so important in cortical development that the remaining allele compensated and boosted expression. But what we did see was in the striatum, part of the brain that's important for many things, but in particular, say movement and language in humans, it was at 50% reduction. And when we looked at all of the behaviors that we could think of, the one that was consistent and striking was a change in these vocalizations. In fact, social and cognitive behavior seemed mostly intact in those animals. And then we did gene expression, and we really embarked on this long-term study of the role of FOXP1 in the striatum. And we could talk about that more. But to me, I think that's another, aspect of using the mice that's potentially a drawback, right? Because I would have expected it to have, potentially recapitulate what you might see in a person, an individual who has, say, a deletion just on one copy, right? Like we think most, usually you're not going to be targeting, you wouldn't have a mutation in both. That would be really unusual. So it was a bit disappointing in that regard. And so that's why in mice, and we know that there's, mice have, especially when we're doing in the model systems, they have lots of ways of kind of getting around how we want to modify them genetically. So they can, what we say, developmentally compensate. And then there's also this idea of, I mentioned at the very beginning, FOXP2 and FOXP4 potentially stepping in and taking over the role of FOXP1. So, why that might happen in a mouse, but not in a human is something that we don't understand. A lot of the work of my lab is to understand human-specific changes compared to other species. And so we're trying to intersect evolutionary understandings of human brain development with like things that we observe when we make animal models, for example. Hopefully that answered your question.
You did. And you kind of touched upon one of the key points that, the human disease is a heterozygote, so one copy is healthy and one copy is mutated. And it's something that we, whenever we read a report from literature, we kind of have to balance that. We have to kind of keep that in mind when we're looking at a model system.
Right.
So maybe a somewhat related question is, so you're seeing these changes and these early changes due to knocking out the FOXP1 gene. So does it tell, what does it say about when you're going to have to do therapeutics? Or does it say anything to the best of your ability to guess?
Yeah, I mean, it makes it seem like it's going to be really challenging from a therapeutic standpoint, correct? But I will refer you to other disorders such as, I'm going to go out on a limb here. I'm trying to remember, I'm pretty sure with Rett syndrome, people have put back in the gene, that's mutated, MECP2, in adult animals and rescued some of the phenotypes. Again, this is in mice. So at least, and they did it in a cell type specific manner. There is evidence from the scientific community and literature that even, and we know that of course that is early in development too, that we might be able to rescue some of the deficits. I mean, for example, the work that Anna showed with the synaptic changes, one can envision that that's something that you could target, right, in an adult brain. And so even if you have fewer neurons, but the neurons that are there, if you can fix the way they're working in some way with a drug, it might be okay.
Yeah.
It may be some benefit.
Right. Yeah, and you're right. I mean, Adrian Birch, one of the classic studies, showed that turning the Rett gene back on recovered a lot of function. It's been shown now for another major autism gene, SHANK3, and for FMR1 as well. But again, those are mice, but that tells, and maybe it doesn't apply, but at the same time, that cuts both ways, right? Maybe what we see, the deficits in mice, may not be fully translatable to humans either.
Right.
So yeah, and I think, and there was a follow-up question, this is from Carl, about, we do, and I think you've already touched upon it. We do, we have to think about, we have to kind of separate in our minds treating and preventing, right? So people look for cures for everything, and in reality, it's about ameliorating symptoms first and foremost, and treating things even after the fact, right? Most of the time, most of our medicines treat things, they don't prevent things, right?
Right. I think also, the earlier question about the stem cell therapy, if, as Nan alluded to, if you could transplant in some sort of progenitor and it would give rise to the right cells that are needed in some way, that's another way to think about it, but that's a long way off.
Yeah. Looking for more questions that might be good now. There's some good ones that might be worth talking about in a panel. So, Jenna, thank you so much. And I really, I don't know if you're watching my social media presence, which I don't have, but I answered a couple of questions in the Q&A. And somebody asked about animals, and I said, you are the world leader in animal studies in FOXP1. So they just had to wait a moment, and you present some interesting data. And as you said, you've been doing this for quite some time, and you have a lot of publications in the field. So thank you, and stick around because we'll have the open Q&A. So you probably all got lots of emails, I know you did, from Rachel Cohn and from Sarah Lynch, and they've really put this all together. And they put together what we did in person, Sarah did a couple of years ago, and I want to thank them. This is never a light, there's always a heavy lift. I really want to thank, the participants. We had a peak of about 75 or 80 individuals joining, and most people stayed the whole time. It's not over yet, but I do want to thank Rachel and Sarah wanted to jump in and say a few words as the next presenter.
Can you see me?
I see you now.
Hey everyone, I'm Sarah. I'm the Communications and Marketing Associate here at the Seaver Autism Center. And like Joe mentioned, some of you might have met me at the International FOXP1 meeting that we hosted here at Mount Sinai in 2019. So hello again to all of you. Before we head into the Q&A session, I just wanted to take a couple minutes to recognize all the tremendous research that has been accomplished since the first diagnosis of FOXP1 syndrome in 2009. And just 12 years later, and from just these presentations alone that you saw here today, there has been some groundbreaking progress. The FOXP1 gene has been and continues to be a priority for our team. And our largest hurdle between accelerating progress for your families is funding. You might be surprised by the high cost of research. It can cost us anywhere from $4,000 to $5,000 to bring in a single family for research, with the travel and hotel costs and the salaries for all the psychiatrists, psychologists, neurologists, and clinical coordinators that they see in the visits. And research requires all the salaries to be covered. Pilar showed results from over 20 families. This cost us over $100,000 to do that research, and to continue this effort, it will cost an additional $100,000 to $200,000 per year. To continue the iPSC characterization that Nan presented, it will cost about $200,000. Anna's work to validate the organoids with Rumi will cost about $200,000. And to translate a biomarker using EEG in mouse models will cost about $100,000. In general, mouse research, like Jenna presented, costs over $100,000 a year. The NIH is slow to fund research for rare disorders, and unfortunately, these advances do not come with small price tags. And until this point, we have relied on funding from the Seaver Foundation and some generous donations from family to complete our FOXP1 research. To stay on the forefront of research and advances for FOXP1 families, we really do need your help. Projects for other rare disorders at our center have been made possible by donations from individual families and from family foundations, including a first ever clinical drug trial for AD&P syndrome. One family sold mask necklaces and donated the proceeds. A family foundation came together and did a walk and run and donated the proceeds to the research done for the rare disorders. Any support from you, your friends, and family can be designated solely to support FOXP1 research done here at the center, or even a specific FOXP1 project like I just mentioned above. We've found the most success in setting fundraising goals to sponsor a project like the $200,000 for organoids, for example. Being able to show supporters what their donation can help make possible is a great driving force. We don't like talking about finances, but the unfortunate fact is research isn't possible without funding. We hope the research covered today and the advances we are on the cusp of making inspire you to take progress into your own hands by making a contribution to the possibilities of tomorrow. An immediate opportunity to donate to our annual fundraiser, which is a video premiere that will be posted online on Tuesday, November 9th, which also includes a FOXP1 family as well. Sponsorships and donations are available and can be directed towards FOXP1 projects specifically. I'll put the link to our fundraiser website in the materials, but it is giving.mountsinai.org/Seaver celebration. And if you have any questions, please feel free to reach out to me. My email address is my name, sarah.lynch@mssm.edu. We'll include that in the chat as well. And without further ado, I'll pass it over to Dr. Buxbaum so he and the other experts can answer all of your burning questions.
Thank you, Sarah. And again, thank you for putting this together. It's a heavy lift. So we are going to have a panel Q&A. I think the panel can turn on their cameras and their microphones. This is the chance to ask all sorts of questions that you may want to ask. I will. I'll take one kind of began with, but I think it'd be worth having multiple opinions on this. So if the syndrome develops in the brain before birth, maybe there's no kind of protein replacement or DNA replacement that would enable full recovery. So it's kind of a follow-up question to what we had before, but I think it might be good to get additional opinions. Jenna told us what she thought and some of the data from Rett syndrome. I wonder if anybody else wants to jump in. It's an important question, obviously. And maybe the clinician, Pilar, you can, when somebody comes in with a developmental disorder at any age, do you say nothing to be done? Is that your?
No, never. I'm sorry. I'm so sorry. I'm having technology issues, so I think
Anna, you wanna give some thoughts on that?
Yes, absolutely. So first of all, I really do not think about drug development as a quest for cure. There is no such thing as cure, I believe, in any disease. There's just the disease management and the improvement in quality of life. So even if the deficits due to developmental delays in utero or in early childhood are severe, I would stipulate that there is still a lot of room for improvement in both the neuronal function and subsequently behavior and the associated quality of life. So intervention at different ages will probably have different outcomes. I think the field is in agreement on that. How much of a differential will depend on specific cases, on specific disorder we're talking about, but regardless of the age of intervention, we hope that there would be an improvement. And I think Jenna mentioned in her talk that intervening at the level of synapse, the connection between neurons, the communication between the neurons is sort of not something that ever expires because it continually happens throughout our lifetime and it directly impacts our behavior. So I believe that there is room for intervention, and there's obviously gaps in our knowledge and understanding of how this would all work in practice. But one lesson I learned through my career is that we learn the most about disorders when we actually try to treat them and when we see the effects of possible treatments. So hopefully that will be the case here as well. And I don't know, Jenna, if you want to comment on, the developmental deficits versus functional deficits in adulthood.
I think you said it really well. I don't have much to add.
Pilar, how's the technical situation?
Yeah, I actually lost. So if you can, I think I know where this question is going, but if you can repeat it for me, I will be better.
Oh, we're talking about, if the insult, so to speak, or the developmental change is, in utero, does that mean that we can't intervene? Maybe, since we did get some answers, maybe you can take slightly different. You already kind of answered it in a certain sense, right? You feel like, maybe you could talk a little bit about how treating attentional deficits might improve other aspects of the behavior. Right? Maybe obviously that's from other studies you've been carrying out, but you already have some thoughts about how to intervene, right?
So I mean, this can sound a little philosophical, and I don't want to get there. But if there's something that I've learned treating many children with neurodevelopmental disabilities, is that kids have kind of unique and their own abilities, and when kids have different or typical developments, sometimes those are not things that you would have maybe discovered or realized if they had gone, quote unquote, the typical route. And I think I always try to think about, like, what are some of those skills and abilities that they have that you want to capitalize. That's one, right? The other aspect of this is that there's two things. One is like, and we really do not know. One aspect is, what do we do when they're younger that will make them a little more steady as they grow older? And this is more important for kids that have atypical developments because their brains are more vulnerable. But this is true for everyone, right? You want to give your kids as much strong support as you can when they're younger, because when life gets a little trickier, You want them to have enough skills to be able to go through life and do that. When kids have developmental disabilities, that becomes extremely important. You want to prepare them for life. So it really doesn't matter when you start, and of course, the earlier you start, the better. But it's very important to kind of go through to do that. And then to answer kind of Joe's just recent comment, you know, every year, every few months, you get presented with different challenges. I think like if a child is getting problems with like executive function, if they have a lot of difficulties paying attention or being present in a moment, or they're getting very anxious, it doesn't matter how many behavioral interventions you give them, they're not going to be able to really internalize them. So treating those things might give them an opportunity to be available and really be present to take part in some of these other things. And none of this, I think the other thing, and some of the parents that have older kids may share that with and I've learned this from the parents that have older kids, right? I feel I learn a lot from the families, is that, you know, the decisions that you make them the best way you can for kind of the following year or the following few months. And then you're constantly kind of reevaluating what is best. But you want to give them the highest opportunity to gain the more amount of skills that will prepare them for life and that will allow them to kind of evolve into the best version of themselves as much as you can. And that, I think it's very important in terms of what decisions you are making. And that is something that can be done even today with the treatments that we have available. And I think that's something that we've learned.
So maybe sticking to this point about, you know, treating things that are manifest later in development. I mean, I think maybe Nan, and I don't know, but certainly Nan, since you haven't spoken yet, right? So you're taking human neurons, you're growing them out, and you're looking at the electrophysiological activity of adult or neurons that are, you know, already communicating with each other, so well past early development. And I would venture to guess that you and Anna would both, you know, if you saw something, you try and actually target that change rather than try and go back in time and target some early developmental change. Is that so? Maybe you can talk a little bit about that.
So, yes, I think
That was a leading question.
to me, because so unfortunately my godson also has autism, but we don't really find a mutation in the known genes. So it's more of an unknown question. But what I learned during all the time that he was getting treatment, and also at the same time thinking what I do may, you know, one day help him, is always comes to the question about plasticity. Because again, neurons are connected, but we don't have a stable brain in a way. Our brain has this incredible capacity to respond to stimulation. The stimulation comes from environment, in you know, communicating with people, but also to any of the manipulations that we can think of. So we do want to know, even after the brain is kind of developed and all the connections are made, do we still have the opportunity to induce more plasticity, or, you know, have the brain being more responsible? And at the same time, other drugs that we can screen to modify the connections or synaptic responses. I think both are possible. And again, I'm open to both options, either using cell transplantation or stimulation, any sort of method to introduce plasticity again into even a well-built brain, or just finding chemicals or drugs that we can do directly to modify the connections. But if you ask me to vote, I am more prone to the plasticity method, finding ways to introduce that, either using cell replacement or drugs.
So, Anna?
I just want to add that and say that, you know, we have to be incredibly nimble in terms of how we develop these treatments and learn as we go. I think we're off to a good start, having all the tools that are sometimes not available for other disorders. We have very strong genetic effect here. We have the preclinical tools encompassing both the cell cultures, 2D, 3D, all sorts of different scenarios, as well as the very well established and studied mouse models. So with all this in place, we should be able to move relatively quickly towards clinical testing and learn more in that space.
So we have a lot of questions and not much time. So Saskia, you answered briefly about DNA repair therapy. Maybe you can give a couple of minutes on that, and then there's a follow-up question. I know you answered a little bit in the chat, but there's a follow-up question I want to ask once you give us a little intro to gene therapy.
Yeah, it's not really my project, so I just typed in the center that Willeke is working on. Can we unmute her? Because she's here in the audience.
Yeah. Give me a second. It's going to take a moment.
Willeke van Roon.
Yeah. Where is that? I'm guessing it's towards the end of the alphabet. So I don't see her on right now.
She might have left.
We can so just maybe
I can try.
Yeah, take it away.
And what would be the question?
Well, so the question you already answered, maybe I'll just repeat what you said, right? So gene therapy is the attempt to put back in a gene that's mutant. So you have two copies, as Jenna said, and one of them has a mutation. And if you put back in more of a non-mutated allele, you might get back to the regular levels of protein. So that's gene therapy. But one of the questions, and this is an important one, about do we need to go to gene therapy? Isn't there shouldn't we be more open to neurodiversity? Right? Shouldn't we move towards a society where in which neurodiversity is valued? That's for somebody living with FOXP1, and that's, I think, it goes back to the question of we kind of talked about before, curing, preventing, but maybe quality of life. That's what Anna used, right?
Yeah, and there won't be a cure in the coming years, but it would be nice if parents have a choice to choose for therapy or not. If it's available, then they can make a choice whether they want the therapy with all the adverse effects or they want to have the best quality of life for the kid.
Yeah, so I think I guess I would say that I think we all embrace neurodiversity. We are unquestionably a neurodiverse group of people right now, but we also know that things can be frightening, scary, disabling, painful, and we try and minimize those so we can have the happiest and most fulfilling life. And it doesn't devalue anybody. It's just, as Saskia said, it's an option, right? Where you say, I have, anxiety disorder, and I'd like to feel less anxious because it is an obstacle. I have obsessive disorder, and I'd like to feel less. I have potential deficit disorder, and I find it impairing in my day-to-day life, and I'd like to treat it. So I think that's where we want to be, right? Where there are options for people, and I do think that if it's, I think that the majority of families that are probably on this call can speak to this, and this is really what the Family Foundation should speak to. But I think there is interest in doing a better job at treating some of the issues that come up with the syndrome. And I think nobody doubts that these kids are wonderful. We've seen lots of them. And we know the families love them very much, and they have all sorts of pet names. One of yours, my daughter's a Foxy Lady, which is a common one we've heard before. So I think that we have to, I think it's an important question, and I think the Family Foundation will really kind of take that forward. So another question from the Family Foundation actually is from Gail Clark. So as was mentioned by Carl, the Family Foundation has started to work on establishing a registry. And I think it might be nice to hear from people. I can go first and then others about what's the benefit of a registry. And I think everything we've heard today in terms of understanding the phenotype and how it may change over time, right? Whether there might be genetic differences leading to changes in behavioral differences, just what Saskia is doing, trying to get a broad look for many people across age ranges and all sorts of things. The registry will be key for that, right? And it's a platform to learn as much as possible about the syndrome. It's not the perfect way, right? Perfect way is, spend 2 weeks in a specialty assessment group and have everything measured and do that every 2 weeks for the rest of your life. But it's a good way to get a quick and broad overview. And then there's an opportunity to reach out to those families and engage them in further studies. Imagine if we said, we think this mutation may be different than that mutation. Well, you go to the registry and you say, well, there are 5 people with mutation A and 5 people with mutation B. So here's an opportunity. So I do think every research endeavor would be enhanced by a registry, and so it's good for the Family Foundation as well. I think Carl just had to jump off. He's got to pick up his kid from school, but I did want to talk a little bit. Gail did ask us to kind of talk a little bit about that. So somebody else, what would you do with a registry? Saskia, this is an easy one for you.
Yes. Yeah, recruitment for my study now goes via the socials and via the diagnosing clinical geneticist, and it costs quite a lot of time. And if I would be able to send it just to you and to say I would like to have this kind of people to be included in my study, can you please send this email to them? That would be a lot easier than it is now.
Right.
So,
and conversely, if there's a bunch of information in the registry, you might change your questionnaires. You might say, "Oh, I noticed that a few percent have this." Somebody just asked about changes in the brain vasculature, right? So you might add that. You might have seen that the registry might describe that in a few cases. It might be part of your questionnaire in the future.
Yeah.
Yeah, I would also add, even for us, it's really important because we want to make connections between what we see in the dish to what actually the patients who carry the mutations have in their real life. Is there a way to do a molecular diagnose and make the connections and finding therapies? That's very important. So I think this kind of registry is just very helpful for us as researchers as well.
Yeah.
I think we'll probably wrap up, but maybe Jenna and Pilar, you can have the final word. You've both been doing this for a while. I think Jenna more than Pilar, but Sarah mentioned how things change even a couple of years. Pilar, how you've been, tell us how long you've been involved in FOXP1 research and what have you seen that's changed? And are you optimistic, pessimistic, or neutral? Again, that's kind of a, I'm going to give you.
I am optimistic, and I'm always optimistic. I think the foundation has been formed, that the families are organized, that there's a thought about a registry. I think that is going to be very important. That will help us, fund research, not just through donations, but then it's much easier to apply for grants with the support of a foundation and to kind of organize and work together. I think also bringing families together from around the world. Let's say we start seeing a mutation in some, I'm just making this up because I think it's much more complicated, but we start seeing that mutations that are around or a certain type in certain part of FOXP1 are associated with X type of clinical presentation, and we see an opportunity for treatment. And then there's a bunch of those kids that are in France and the Netherlands or Australia. We can actually communicate with them and kind of work with them together. I think now we are, despite the setbacks of COVID, I think it has also taught us that we can do this from all around the world, right? Where Saskia is like at 5 a.m., I think, here with us and sharing, but I think that's something that we have to leverage.
So Jen, you're gonna get the last word, but I put up a slide and I probably shouldn't have. It's unpublished by other people, but it has been presented. But this is the newest data from 60,000 individuals with autism. And FOXP1 is 1, 2, 3, 4, the 7th most common mutation in this cohort. So somebody asked how frequent are FOXP1 mutations. And so even though it's a rare disorder, there's a lot more out there. And one of the things that Pilar pointed out is if you go to the NIH right now and try to get funding, and there's no, there's no register. You can't say there's 100 people who could participate, or, there's 1,000 people that are suffering right now in America or in Europe. It just doesn't become a priority, even though we know that FOXP1 is pretty common amongst the rare genetic disorders. Most people don't get genetic testing, right? And so even just the Family Foundation getting the word out and getting registry information is a game changer. So anyways, Jenna, sorry, Jen, we have Jen here, so I'm calling you Jen.
Okay. We'll save discussions of NIH funding for a different call on a different day. But
all being recorded, so knock yourself out.
But like Pilar, I mean, I am the ever optimist. And I think back to when I first started thinking about working at FOXP1, where there were just maybe a couple of papers on it. And, wasn't, there was no link to, it was more studied as just another gene, important in development, right? It wasn't had anything to do with any patients. And so individuals. And so I think we've made huge progress in a relatively quick amount of time. And what I'm most excited about to see coming down the pipeline, all of the work that I've been seeing from all of you, and how the work in my lab intersects. So how does the mouse and the human data converge, and how we can use that to think about therapies. And so I'm just looking forward to the next few years and really making huge progress on that.
Well, thank you all. We have another question or comment from Amanda, who said they are very grateful for the work of this group and all those represented in the presentations. And thank you for your time, for the information you're sharing, and for everything you're doing. So I just want to thank you all for presenting and for everything you've done on behalf of myself also, but also Amanda and all the participants. And we'll send out information for as we promised. And there'll be more of these events, but certainly now that there's ongoing Zoom meetings through the Family Foundation, that's an opportunity to catch up, and we would be happy, I think everybody here would be happy to present in those forums at different times. And as Carl mentioned, there's a commitment to make another, hope was to be in person, but maybe not, big meeting early in the first half of 2022, and we hope to see you all there, virtually or otherwise. So thanks to all the speakers, I really appreciate it. Thanks to all the participants who kind of stuck in here for many hours, and thanks for those who are actually not on the North American continent, who had to stay up very late or get up very early. So have a happy fall, and all the best.
Bye. Thank you.